Hello, and welcome to the Neo4j Healthcare and Life Sciences workshop. You should be able to hear and see three lovely people. Michael Hunger from Neo4j, our head of developer relations, Martin Preusse from Preusse and Partners, and myself, Alexander. I'm just here to facilitate a little bit, but I think we had slight issues with sound, but now I think both Martin and Michael are able to say something, and you will be able to hear.
I'm able to say something.
Okay, good.
I'm also able to say something. I was kind of waiting for you because the plan was that Mike is done, so I was just waiting.
Sliding back again. Michael, you are muted.
One, two, three. This is better,
no,
right?
That's perfect.
Okay, sorry for that. So I hope you can see my screen as well. Martin, can you see my Alex?
Now we can see it.
Yes?
Mm-hmm.
Super. So very welcome, everyone. I'm super excited for the third Healthcare and Life Sciences workshop for Neo4j, running now in November 2021. And I'm especially excited because I was part of the first two workshops as well. The first one was already almost 9 years ago in Ghent, where we had 6 talks and 1 workshop. And then in Berlin in 2017, so that was 4 years ago, where we had 11 talks and 3 workshops. And we also published the proceedings there. Both of those workshops were amazing, really great interactions. I learned a lot, not being a biologist or biochemist myself, so I learned a lot of details. But I was especially impressed by the strong collaboration, the very positive atmosphere, and I hope that we get the same today as well, and also by the deep talks. So it was not just scratching the surfaces, but really talking in detail about what people are doing with graphs, and that's really amazing. So, and just to give like a high-level overview over that, so we've seen people using graphs in life science in all kinds of areas, from drug repurposing, cancer research, proteome networks, reactome networks, any pathway analysis. We've also seen people managing clinical trials, patient groups in graphs. Of course, analysis of scientific papers, both just citation networks, but also NLP and detailed analysis between papers. We've seen last year COVID graph, which we'll probably hear a lot about today as well from Martin and others. Drug optimization, and also healthcare education in general. So there are lots of areas. I'm probably also scratching the surface here, and it's something that I'm really, really excited about. And because I also see the 21st century as the century of life sciences and making life better for all of mankind or humankind. So that's really great. Of course, there are lots of challenges, right? So many people that work in these areas have never heard of graphs. They've never used them, seen them. They know what a network is, but they've never used graph as a tool as such, right? So for their data. So they have to really learn some tooling, really learn some modeling. Most people still come with spreadsheets and relational databases and a bunch of Python code perhaps, but I think there's much more potential there, kind of by looking at your data in a different way. And what I also am really looking forward to is just have better collaboration. Of course, we have different research institutions collaborating, but also on the data level, right? So we don't need to reinvent the tools and the data and databases and data sources. We just should work on bringing them together. Everyone focuses on their specialties, and then we can more share and reuse things that have already been built and such. So which is about getting data in, publishing data, querying cross data sources, and so on. Of course, ontology play a big role in life sciences, so that's something that we also are getting better at. Data quality, of course, especially if it's real-world data coming from clinical trials or inputs from healthcare authorities and or hospitals. So there's data quality also in question. Or if you have different data sources, how do you kind of match them with different terminologies or different, values there. And then one area that's super exciting is the whole data science aspect, right? So how can we use and apply and utilize data science methods, so machine learning, social network analysis, graph algorithms, and things like that on top of that data, so that we can also use the context of things that are there. But I think it's a really big opportunity for all of us, because as you all know, real world structures and processes are networks, right? Look at a protein, look at a human pathway, look at a genome, at a cell. There's all these kinds of processes, and look at the brain, neurons, and so on. So everything is basically pieces of information that are connected or working together, processes that are working together. And a graph is a really valuable tool to represent these things and make them accessible and visible as well, right? So we can use this not just for storing the data, but also for visualizing it, for exploring it, for querying it, for finding patterns, and also for educating others about it as well, right? And so the opportunity here is really kind of to take the existing data, turn it into a model that's so much more suited for it, and then bring it together and collaborate on a large scale across the scientific community as such, right? So, and for that, I would love to see more public databases that are open and accessible and that are integrated, so that you can use that on your research or on your projects, and you don't have to spend so much time on all the base interactions and getting data in and so on, because if someone else has already done it, then why should you do it again? And which also means pulling data together to form larger, more complex and more comprehensive models of reality as well, right? So I don't just want to look at genes. I also want to look at like cell processes. I want to look at expressions. I want to look at local inhibitory networks and things like that, and these kinds of processes. So I really want to see kind of how are all these kind of things coming together and working together, for instance, on a certain expression, a gene expression, or a certain disease, and or a certain drug mechanism as such. And what's also interesting is, because the data volume grows, especially as we know in genetics and other areas where we capture a lot of data, genome analysis, and so on. So there, it also becomes more important to have unsupervised learning methods. So you can't sit there and label everything. I mean, we've seen it with AlphaFold already, so that there are mechanisms in there that allow computers much faster to recognize patterns, to identify certain repeating processes or flows as well, and use that more data to kind of label data, and then we can use it later on. Of course, that's kind of not just pure machine learning, but it's human in the loop. So basically, it's a good collaboration between both as such. On the graph side, especially also in Neo4j, there's also lots of things happening. Since we had the last workshop in 2017, we released 8 versions of Neo4j. There's lots of innovation. There's many more companies have entered the graph space, so you don't just have Neo4j as one technology provider, but many others as well. There's a lot of standardization work around GQL going on, so hopefully in 2022 we'll see the standard, and then the first implementations coming out that's heavily based on Cypher. We have cloud services like, for instance, Aura DB, that allow you to publish your data to the cloud and then have other people use it without you having to take care of management and operations of those databases, because that's something that you really don't want to spend your time on. The Graph Data Science Library, which started as the Graph Algorithms Library, has now become something that's part of the product. It has a lot of graph algorithms that perform really well on large data sets, but it also supports a lot of graph embeddings already. So you can basically compute embeddings for your entities as vectors that you then can use to train your ML models either inside or outside of the graph. It now has also machine learning pipelines already. So you can already integrate this really well with things that you want to see, very contextualized information, and take this information basically into making more sense of your data. On the ontology RDF level, there's the Neo Semantics Library, which can at least input lots of different formats, make data from a property graph in Neo4j available as RDF as well, and allows basically better integration of all kinds of data sources as well. And speaking of integration, there's also lots of work happening on the connector side. So we have a Spark connector, which now also supports Spark streaming and which you can use from Python, from R, as well. Kafka integration for streaming data. So if you're getting data from a large source of, like, for instance, clinical trials or healthcare authorities, then you can just put it into an event stream. And of course, JDBC, ODBC integrations for things like Tableau and other visualization tools as well. And if you're going to the cloud, then of course, all the cloud providers have massive ML pipelines there for NLP, from image processing, image recognition, labeling. You have existing data sets that you can integrate with. So there's also many more potentials than running it on your own local data center or infrastructure. Putting things into cloud, it enables also so many more integrations and combinations as well. In Neo4j, we also for federation have Neo4j Fabric, which allows you to basically easily connect or query across a number of different databases with different data models, or also shard out large volumes of data as well, and then query in a federated manner across those. So that's also one first step into the direction of not having to move all the data into a single database, but basically supports federated queries across a number of databases as well. And then with things like Bloom or Graph History, for instance, you can do better graph visualization at scale, where they use WebGL or OpenGL mechanisms and GPU compute to visualize tens of thousands or millions of nodes and relationships, to see also patterns visually and see flows visually as such. So there's a lot of stuff happening. I know many of you are also interested in these technology aspects, so we'd also love to help more. So for this aspect of better collaboration and doing more, we'll have a survey after the workshop, and I'd love you to fill out that survey to give us also input. What are you needing? What are you looking for? What kind of workshops, trainings are you interested in? And so on. What kind of collaboration opportunities do we have in this space? We also want to update our life sciences page, so Martin will help us with this a lot. And then we want to have more workshops of this kind, like today and tomorrow, on a, let's say, yearly basis, so that it's not as rare as every 4 years or so, but it's on a yearly basis. And then, of course, if there are projects that are really impressive, that are public, that are open, helping the world get better, then of course we also highlight, award them, and support those projects as well. So that's at least my heartfelt wish, that we go from here to a way better area of collaboration as such. So I want to encourage you to enjoy these two days of knowledge sharing, listen to the talks. I'm really excited for all of them. Deep practical talks. Big thanks to all the speakers for taking the time and preparing and offering to speak. All the sessions will also be recorded, and we'll see them later. And big thanks to Martin and Alexander for organizing this workshop. Without you two, we wouldn't have this amazing event today. And with that, I'll hand over to Martin. And I don't know, Martin, do you want me to advance, or do you want to share your screen instead?
I will share my screen.
Okay, cool. So I'll stop sharing.
Perfect. You should see my screen now.
Yes.
Fantastic. So thanks for the introduction, and it was a pleasure, of course, to organize this. Like you said, it's all about collaboration, connections, and network, right? And in the next 10 minutes or so, I would like to briefly walk you through our agenda. But before that, I would like to kind of go a bit into detail of what kind of collaboration I think is important in our space. So the first axis of collaboration, if you will, is along the value chain of pharmaceutical research and healthcare. So what I see a lot in real-world Neo4j projects and real-world knowledge graph projects is that, you know, having data connected really helps people with different backgrounds to come together. I mean, there's a long value chain from the initial basic research that we do up to a point where we manage to, you know, develop a new therapy, develop a new drug, and bring it to market. And then, of course, collect real-world evidence and then follow post-market developments. And I think more collaborations between the, let's call it, life science side and healthcare side is something that will be incredibly valuable. And I mean, this kind of general drive towards RWE is essentially to force us to do that. And that's one of the reasons why we try to get talks from different stages of this value chain in our workshop. Another collaboration that I think is super important is the public and private sector collaboration. There's so much overlap between the research that happens at universities and the basic development, drug development that happens in the pharmaceutical industry. And the same, of course, is true for medical informatics and healthcare. And there's so much overlap, but I think there's really room for improvement in terms of collaborations between public and private sector. And for me personally, one of the most important aspects of Neo4j is the fact that it's a tool in the end that can help people work together. In the end, for me, one of the main reasons is not about query performance and all that. It's this visual aspect, the fact that your data is tangible and more visible, and that helps non-IT people come together over the data. And I mean, the data is obviously the data that we collect in primary healthcare is obviously very, very valuable for drug development and basic research. So bringing these sides together, I think, is super, super interesting, and one of the most important aspects for me. In our workshop, we'll have 13 talks in total, and I would like to briefly walk you through our agenda. We are obviously in the middle of the opening, and the first talk today will be from Novo Nordisk. So we'll start with pharmaceutical research, and this talk I think is very interesting because it has a strong focus on graph data science. And Michael already mentioned that since our last workshop, so many new technology elements joined the Neo4j ecosystem. The official graph data science library, but also more architecture and infrastructure-oriented things like Aura and Fabric. But graph data science is obviously a very, very interesting element, I think. And the first talk will have a strong focus on graph data science and analytical methods. The next talk it jumps over into the healthcare area. And this is very interesting because this is about connecting Neo4j, the knowledge graph, with data from the UMAP Common Data Model. And everyone who works in healthcare knows about the UMAP Common Data Model, I suppose. And activating OMAP data in a Neo4j graph can be super powerful. And after that we'll have a talk, a more application-oriented talk. Federico Braga from LifeGen will talk about their technology stack and how they build web applications for healthcare using Neo4j on top of Neo4j. And what I really like about the first slide of our program is that we already have researchers from Northwestern University, a big pharma company, and a startup from Europe. So bringing together these different areas, hopefully fostering collaborations between researchers, big companies, small companies, and so on. I think this is really at least for me, one of the main goals of this whole workshop. Later, we will continue with another talk from the pharma area. Jérémy Gagnard from Servier in France will present their Pegasus target discovery platform, which is also super interesting project in terms of data integration and so on. The next talk, I will come back for the next talk, that will be about Health Echo. Health Echo, I don't want to promote my own talk too much, but Health Echo is an open source and non-profit organization that we started around the COVID Graph project. I hope that at least some of the people joining here know about COVID Graph. It's an open source knowledge graph, Neo4j knowledge graph. We just collected a lot of data about COVID, and that started last year, and we now have a huge community around that. And we started this non-profit organization, Health Echo, and it will always stay open source and non-profit. And our goal is really to develop it into an organization, into a community where we can all come together and contribute. So I'll show a bit of the background and of course the outlook and goals that we have with Health Echo. We'll continue with more molecular biology. The next talk by Claire Simpson from Cell Signaling Technology is a published paper, published earlier this year, I think in May. about analyzing perturbed co-expression networks in cancer using graph database. So what I really like about this project is that it has a publication, of course, so we can look at all the details, and it has a very interesting application that was really described from beginning to the analytical application in the end. And the last talk for today is also going to be very interesting. It's something I've been looking forward for a long time. Evelyn Travnik from the Novo Nordisk Foundation Center for Biosustainability will present their Lifelike platform. And the interesting aspect here is that they have a massive data integration engine behind this platform. So this will be a very interesting talk. I think a lot of software that they develop is also open source and potentially reusable. So this will be a very interesting talk. And with that, we're going to close for today. We're going to continue tomorrow, starting at 10 a.m. Central European Time, 4 a.m. Eastern Standard Time. We'll have a short opening, of course, and then we are going to have a last-minute talk. The agenda was full, but we had to squeeze this talk in because it's also a really interesting project. Rita Kolako will talk about a graph-centric framework for translational clinical proteomics. And this is really interesting because it's also public. So it's a public knowledge graph. There's a bioRxiv paper on this, the clinical knowledge graph. And she will present, at least in a short presentation that we managed to squeeze into the agenda. She'll present the clinical knowledge graph. And they really have a lot of super interesting applications for this. And the good thing is it's a published work. So maybe some of this might also be reusable. We continue with more public research. Alexander Jarasch from the German Center for Diabetes Research will present a couple of use cases from simple queries to advanced algorithms that they developed on their pharmaceutical knowledge graph. This is a really interesting project because it's a massive graph with a huge focus on collecting data from publications, from PubMed, and so on. So this is really interesting. With the next two talks, we walk away a bit from humans and diseases and focus on microorganisms, which is obviously also very interesting and very related. So Xixing Huang will talk about Neo4j for bacterial genomes. There is a series of blog posts about this topic that he published already, and I'm really curious to see how he models genomes and what kind of queries he can run and do for bacterial genomes. The next one from Piyush Sahu from Clariant is also about microorganisms, and he will essentially describe how he integrates multi-omics data in a knowledge graph and what kind of queries and data analysis you can do on this integrated multi-omics data. And I think this is really interesting because, I mean, we all, at least in molecular biology, we all work with multiple omics levels these days, right? I think it's just very common, and that's a very interesting talk. We continue with more public research. David Motin from Aarhus University is a graph analysis specialist, and he will present a couple of methods about unveiling the knowledge in knowledge graphs. I really like this title. And the interesting thing is there are so many graph analysis methods that are available. And while the Neo4j Graph Data Science Library is very good and it's growing and developing, there is so much more in terms of graph analysis methods. And David definitely knows all of them. So this is going to be an interesting talk, and I think also an interesting way to collaborate. We go back to healthcare with a talk from IQVIA. Sasha Gowey will present a Neo4j database that is used for evaluating drug safety. And this is definitely interesting, and I think somewhat related to the talk from Northwestern about the OMOP data. The last talk tomorrow will be about clinical knowledge graphs with Spark NLP on Neo4j. And what I like here is that NLP is obviously in the whole medical informatics space incredibly necessary technology in the end. There's so much knowledge hidden in doctors' notes, essentially, and health records. And activating this and getting this into a knowledge graph is, I think, something that is incredibly valuable, to get more data into the hands of the people who know what to do with it, and to get some useful output. Tomorrow we will have a closing session. And like Michael already said, there's so much room, essentially, for collaboration and so much overlap. And we'd like to take the closing session tomorrow to really focus on this and discuss with you how we can proceed, what could be the next steps, and what kind of collaborations could we start, and where could we reuse data models, reuse data, and so on and so forth. Because I think there's really a lot that can be shared in a way that everyone wins in the end. Okay, let me mention a few organizational things our first session today. So if you're watching via Bitmarker, please stay in this session for the whole day. So on the Bitmarker website in the beginning, there were individual sessions to show you the agenda, but we will stream into this session for the full day. And there's another session coming tomorrow. But stay in the session for the full day. And like Michael mentioned already, there are so many technology aspects in the Neo4j ecosystem, GDS, Fabric, Neo-semantics, which is also vastly improved these days. And we would really like to get your feedback on what kind of technology you want to learn about, because we would definitely be open to do follow-up workshops, etc. But the most important thing, I think, is, like Michael also mentioned, I definitely want to turn this into a yearly event. This year we are fully remote, but I really hope that this can be an on-site event next year. Let's see how the COVID situation develops. But we will definitely have a yearly event because there is so much overlap, and we're going to see this during our talks, and we just have to work together. Okay. That's pretty much it from my side. I'm really, really, really curious about all these talks. I'm really excited. And with that, we can move to our first speaker. Lisandra Mendoza from Novo Nordisk will present her work on graph data science and knowledge graphs for target discovery for cardiometabolic diseases. And Alex, I think you can switch over now to Liz.
Yes, hi. Let me just share my window. And of course, because I am the first one, I have some issues finding the window.
We will find it, no worries.
Okay, I think I'm just going to share the entire screen, and then from here, I will just bring up the presentation. Here it is. Can you all see it?
Yes, we can.
Okay. Yes, perfect. Okay, well, first of all, thank you, Neo4j, for the opportunity to talk about how we at Novo Nordisk Research Center Oxford use knowledge graphs and graph data science for the discovery and evaluation of novel gene drug targets for cardiometabolic diseases. I will first explain why at NNRCO we care about knowledge graphs. Then I will explain how we build our in-house one, and also describe some of the applications that we have developed on our knowledge graph. Okay, so first, just a little bit of a background about Novo Nordisk. Novo is a Danish global healthcare company, most well known for their insulin production, but we also have efforts going on the areas of rare diseases and cardiometabolic diseases, including diabetes, obesity, fatty liver, osteoarthritis, and others. Novo have various satellite sites, such as the one in the UK, where I am based. Now, first, I will give just a quick intro to the basics of property graphs. So in a graph, we know is a node and an edge that connects another node. And if it's a property graph, we can have properties stored either on the nodes or on the edges. And I will just focus on the use of graphs as databases, where instead of using the rows and columns of a table, we use the nodes and edges of a graph. You can always turn a table into a graph relatively straightforward, but turning a graph into a table can be much trickier, depending on the complexity of the connections between the nodes. The advantage of graphs is that they are super flexible. You decide what bit of information you want to have as a node, as an edge, or as a property. So you have tons of data management power. What is super useful and unique to graphs is the topological information, meaning the information of what is connected to what and what are the characteristics of these connections. Now, what is the difference between a graph database and a knowledge graph? I think it's a little bit debated in the field still, but basically it's that a knowledge graph is a graph database enriched with information from a particular field, and experts of the field were involved in the modeling of the data and in the careful curation of the data that is ingested to the graph model. The point of using graph data science is to find relationships and combinations of diverse data. So to your graph modeled data, you can apply various graph-based algorithms, which you can generally classify into 6 categories. First one is community detection, aka clustering. Then you can also evaluate the centrality of a node to give you an idea of the importance of the node. You can also evaluate the similarity between 2 nodes. You can also do link prediction, so estimating the likelihood that 2 nodes that are not connected could actually form a relationship. You can also do pathfinding and searches, and then finally you can also do node embeddings to evaluate a node by the nodes in its neighborhood. On their own, these algorithms can give us relevant information and insights, but we can also use them to do some feature engineering, meaning to create features to append to our data to be fed into another machine learning model. What is the usual complaint you hear from a data scientist? It's, oh my God, I don't have enough data to train a good model. Well, a data set enriched with topological information is expected to allow the model to make better and more accurate predictions. So in NNRCO, we have used GDS algorithms with these 2 approaches, feature engineering and direct graph extraction of insights. I will talk about those things in the next slides. But first, I'll talk about the components of our knowledge graph. Since we have a disease-focused knowledge graph, the schema is disease, gene, and protein centered. Our knowledge graph is made of 3 components. The first component is the gene protein general knowledge. So it has available public and in-house information on gene and protein annotations, interactions, associations. So for example, pathways and protein-protein interactions, gene ontology, biological processes and cellular locations, protein complexes, compounds bound by proteins, etc. It also has information on GCC associations, obviously, like proteins targeted by a drug, also information coming from clinical trials, from genetics, and also information around human diseases. So for example, we have the human disease ontology, the human phenotype ontology, and other disease-relevant ontologies ingested in the graph. This component is like the backbone of the knowledge graph. Then the second component is the one coming mainly from tailored text data analysis, aimed at enriching the graph with information for specific diseases, the diseases that nobody's interested in. I will talk more about this enrichment process in the next slide. The whole text sources that we use are papers from PubMed, including a special full access to Nature and Science Direct, then also NIH grants, patents, and pharma news and conference abstracts as well. And then the third layer comes from novel-specific knowledge. For example, information on genes previously or currently being investigated as druggable targets for our diseases of interest. Also, information of the assays we have in-house for the validation, the readouts, the transcriptome results from those cells, etc. And the time and effort needed for the development of each layer varies due to various factors. So for example, the workflow to build the first component was first bulk download of the data from various databases, then lots and lots of data wrangling and harmonization of annotation and IDs. So actually a fair amount of manual creation was needed for some of the databases. And then a schema was designed to fit all the data. And then of course, that model went through rounds and rounds of design and redesign. That took some time. But the enrichment of the diseases is actually taking much longer. And to ingest the results, some modifications to the backbone data model are sometimes needed. And then the integration of this last component is not a bulk data ingestion. It is more of a manual addition, one by one sort of thing. The components of this slowest growing one are the slowest one, but they are arguably the most valuable ones for our purposes. So in total, we have around 45 different data sources, some public, some private, some coming from extensive curation efforts over public or internal sources. And to work with all of this, our workspace integrates Neo4j for the database usage and the GDS they provide. Then we also use Bloom for the visualization and some Python as well for more complex data wrangling and other ad hoc data science approaches. All right.
Sorry, Marie.
So for the second component,
there's a little window covering parts of your presentation which says, is sharing your screen. Do you mind clicking? Yeah, perfect.
Oh.
Super. Thank you.
Sorry about that. Okay, yes. So for the second component, as I said, we use text data approaches for targeted enrichment of the knowledge graph. There are two main approaches for analyzing text data at scale. One is NLP and the other one is text mining. So NLP works with semantics and linguistics by using graph models and machine learning models like neural networks. And its main concern is to understand what the text means. Then text mining works with keywords by using string matching and statistical models. And its main concern is to extract the specific information that you need or that you want. But then another way of collecting text-based knowledge is through manual curation and the use of ontologies. I've mentioned ontologies in my previous slides, but what ontologies are is what they do is they standardize the meaning of a word for a given concept in a given field and allow you to work with concepts and relationships in ways that are close to the way humans perceive interlinked concepts. So we are using all these three approaches for our disease enrichment. We are working with other parts of the organization to search whole texts using NLP models and text mining pipelines and ontologies. And then we're also working with a company that performs literature manual curation. I will describe in a little bit more detail the enrichment approach based on curated data that we do. So we first define a list of disease hallmarks, which are phenotypes that are relevant to the development, identification, or progression of a disease. And by phenotypes, I mean conditions, metabolic signatures, symptoms, etc. Then that list is manually annotated by a curation company that we work with, and they have their own pipeline for selecting which papers to read based on the keywords that we provide to them. Then from these papers, they extract information about gene disease associations, such as the directionality of it, and then also when available, they extract any clinical details, like the experiment, the patient characteristics, and the treatment outcome. Then we use some tools from the Novo Nordisk Ontology department to map some Novo-tailored ontologies that we have onto the curated results. And then we can also extend such ontologies with unmapped curated terms. And then once all the results have been unified, we add them to the knowledge graph. We have done these hallmarks for obesity and osteoarthritis in the context of obesity. And after we added the curated data, we identified a fairly large number of new gene disease associations, enriching the disease knowledge graph, allowing us to explore a larger space in the graph when looking for potential targets. And then the next curation steps that we have planned are to be focused on fatty liver and atherosclerosis. Okay, so now I will tell you about the uses that we have been giving to the developed knowledge graph. So far, we have implemented 2 published graph-based methods that we have modified and tailored to use them for drug target identification. There are a couple of other implementations we would like to do. That is why I'm not marking this issue as complete yet. And I don't think it will ever be complete, though, because there are so many different ways in which you can work with this that there's always some next project coming up, I believe. And then the second and third application are for target novelty evaluation and target prioritization. These applications were developed because usually the result that you get from an in silico analysis is a long list of score targets. So then you face the questions of where do you put the cutoff threshold, how tight, or how do you select the ones to move forward for in vitro validation in the lab? And so these applications are meant to help us with that. Then the last application, which we have not developed yet, but is in our backlog, is a safety evaluation metric by using the data in the knowledge graph regarding side effects, for example. Okay, so we use our knowledge graph to automatically quantify the amount of information there is on a target. Increasing novelty means decreasing information in the knowledge graph. The different aspects of novelty that we are most interested in are the following 3. First, is it associated to my specific disease of interest? Or is my target associated to any other cardiometabolic disease? Or to any other non-cardiometabolic disease? The second angle is this target already targeted by a drug, or is it already in clinical trials for our disease of interest, or about for any other cardiometabolic disease, for any other non-cardiometabolic disease? And then the third angle is, just in general, how much is known about this target? So for this last point, we calculate a connectivity measure called degree-weighted path count, or DWPC. So what DWPC tells us is how connected is one node to another node by traversing different defined paths. And you downweight the paths that go through highly connected nodes. And then for the calculation of point 2, we use the scores reported in Open Targets, which is a public database that we also have ingested in our knowledge graph. But for the calculation of point 1, this one, we use a literature-based gene disease association score. And that one is a score that we have ingested into our graph as well that comes from one of our vendors. And like I said, the level of novelty we might be interested in varies depending on the purpose of the project. So instead of merging the scores from these 3 different angles, we created 8 different levels of novelty assigned by this simple decision tree that we have here. So if the gene already has a drug targeting it, or if it's already in clinical trials, then it has a novelty level 1. On the other hand, if there is nothing known about the gene, then it has novelty level 8. And then you might also be interested in comparing the scores of the different angles amongst a bunch of genes that you're interested in, say your top favorite ones from your project. You can easily do that with a radar plot like shown in here. And then, in order to get some rough preliminary idea of the importance of a gene to a disease, We consider all the available information around it using a combination of 4 GDS algorithms from 3 different kinds: path search, centrality, and clustering. The first algorithm is the shortest path between a gene and a disease of interest. The shorter the path, the higher the score, because we take the inverse of it. And the second metric is similar, this one, but instead of looking into the shortest path, we look into the first n shortest paths. So this allows us to distinguish between genes and diseases that are tightly connected and those that are a little bit more loosely connected. We also use personalized PageRank to allow us to know how important a gene is from the perspective of a disease node. And then the last metric is based on the Louvain clustering. So we perform several rounds of clustering and calculate how frequently it is that the gene and the disease fall within the same cluster. We integrate the results in such a way that the more frequently the gene node and the disease node share a cluster, the higher the score. And then finally, we combine the 4 different scores into a single score to help us rank the list of potential targets. And to finalize this section, I will just briefly mention the 2 methods that we have taken from the literature and implemented an in-house modified version of them to use for gene target discovery. The first one was HEDIANET, which learns which types of compound disease paths discriminate treatments from non-treatments in order to predict the probability of a compound treating the disease. So we just modified it so that instead of looking at compounds as the main node of the path, we look at genes. Briefly, it consists of the following steps. First, we define a set of paths in the graph that go from a gene node to a disease node. Then a degree-weighted path count is calculated. This is the same DWPC metric that I mentioned in the slide about the novelty score. And then we randomize the network and calculate the DWPC on the paths, again, on this randomized graph. Finally, a logistic regression model using the DWPCs is developed to identify novel genes as associated to a disease. And then the second approach is this one called MindWalk. This method works by first defining the start, the length, and the end of paths, where the root, meaning the first node in the path, and which we make to be a disease node. And then at the end, we make it be a gene node. The disease nodes have labels to group the disease together. So for example, cardiovascular disease, metabolic syndrome, or diabetes complications. I mean, the labels depend on what you're looking for. And the genes are not labeled because those are the ones that we want to classify. So then you iterate over every root and store the neighborhood of the defined length as a list of sets. And then you calculate the information gain, or IG, of each path in this list of sets. The IG is calculated as a weighted reduction in entropy obtained by partitioning the data to calculate the prior and the conditional entropy based on the presence of the unlabeled gene node. So it is conditioned on the walk. And then use the information of the fraction of the labels in a given partition dataset. And so in order to calculate the IG of a gene, its presence is tested in all these sets of neighborhoods. And then the label of the path with the highest IG score would be the one assigned to the gene. We have applied all our different methods for the discovery of targets for atherosclerosis and fatty liver. And the next steps for us are in vitro validations. We have different assays in NNRCO to test for some fatty liver phenotypes using primary human hepatocytes, and also some assays for atherosclerosis using endothelial cells. Then we also have some other more specific assays in another of novel satellites that is in China, and then some much more detailed methods in the headquarters in Denmark to go down to investigating the mode of action of those genes that pass all the rounds of validation in Oxford and China. And just like I mentioned in the section about the construction of the knowledge graph, using a graph database gives you tons of flexibility for the modeling of your data. You can add more data from other data sources, change the initial data model, update it, upgrade it, you do with it whatever you want. But with great power comes great responsibility. So, for example, this is the evolution of our data model. When we first started building the first component of the graph, it looked like this. And then after 3 months or so of working on it, it looked like this. So it's much more slimmed down and structured. And then by the time we finished all the first component and adding a portion of the third component, it looked more mature, like this. It was to this model that we applied the drug discovery pipelines I described before. But then, after we ingested the text-derived disease enrichment data, the data model exploded and went slightly out of hands. So what we're doing now is going back to the drawing board and polishing the schema, refactoring the graph to remove redundancy and to optimize for specific kinds of queries that we now know that we need. So my main take-home message, really, for anyone thinking on building their in-house knowledge graph is to keep the data model in check at all times. And with this, I finish my presentation, and thank you for your attention. If you have any questions, I think we have some time for a couple.
Yes, so thank you so much. That was a really interesting presentation. And we have some time for questions, and we do have a couple of questions. So let's start with the first one. A really nice question. How much work was there in getting all this information ready for computation? So I think you're muted right now.
Yeah, am I muted?
Sorry. So the first question was, how much work was there in getting all this information ready for computation? And this had a follow-up question. How large is your team to do all this? So how much work did you have to put into this?
Yes. So some of these components were already in a database that we have in-house that already has gone through the work of collecting and collating public databases. And we have an API. So I just use the API of our relational database and pull everything. So that took, I don't know, let's say a month. What took most of the time was the harmonization of all the pulled sources, because a lot of manual intervention was needed for some of them. That one, let's say, added 2 more months. In total, I think I had a decent schema, something. I got to this after about 3 or 4 months, and then we got to this after 6 or 7 months. And the group that I have, we have students doing short-term rotations with us. So we had someone with us for 6 months, and then another one with us for 3 months. So the group is a little bit floating, but we are, like, I'm the one coordinating most of the efforts.
Mm-hmm. Okay, so I think that should answer that question. So the next one is about public data and public data sources. So you are muted, I think.
Oh, am I?
Yes.
How did that happen? Okay. Can you hear me now?
Yes. So the next question is about public data and public data sources. So there's the question, are there any restrictions for using public data in your projects?
Yeah, we definitely check which ones we have access to and which ones we don't. There are some which we don't, and those we exclude. There are some that maybe we had the license before and now it has expired, then we either remove them or make sure to not include them. We are careful with that.
Mm-hmm. Okay. And if I'm allowed to ask, I would like to ask a follow-up question. when you integrate all these, like the first column that you showed, all the basic data on genes and so on, because we are here, and I will come back with these questions all the time here for collaboration. Do you think the data model of this, and your data noding and all of that, do you think this is something that you could share, or would you consider that to be something that has to stay internal, if I'm allowed to ask that?
Sorry, so your question was whether it is updated regularly with the same scripts, or if I need to modify the schema?
No, the public data that you use, and the data loading for the public data, and the schema for the public data, is that something that you would share with the community in principle? How would you approach that? So
Yes.
the way how you work with public data in your knowledge graph.
Yes, we do have a strong support for a strong push for publishing as well. So we're actually using the knowledge graph also as a support for another project that is also focused at prioritizing targets using different things, and the input from the knowledge graph is expected to be one of those layers that are married together. So when and that one we definitely plan to publish. So when that is published, the knowledge graph will also be published.
Mm-hmm. That's interesting, I think. So there's another question on Bitmark in the chat. Thomas asked, how hard is it to combine various medical databases in a single graph?
Very. I think there really were tons and tons of hours of discussions with clinicians and other people from in silico and other biologists as well to get to the model, to the data model that we have right now. And still we every now and then change it, especially when we add a new data source, we change it.
Mm-hmm. So is it hard or is it not hard? Is it hard or is it easy?
If you have a good communication channel and you can actually get with a biologist in the same room, and you definitely need a whiteboard, then it's fun. Not difficult or easy, but fun.
Mm-hmm. Okay. Let me check the Q&A. There's another question coming up from Umberto. He's asking, how are you modeling gene-protein complexes with your schema? Yeah,
that's a good question. So we have a node that is a complex node, and then we have the proteins that participate in the formation of that complex. So we put a link between the protein node and this complex node for all of those.
Mm-hmm. So you have complexes as an entity in the graph, as a node type, and then you map them together.
Yeah.
Okay, that's interesting. Let me check the chat if there are more questions.
Then also I can elaborate a bit more on that. We do a similar thing for pathways and disease pathways as well. And for the pathways, we also went through the step of curating them and annotating, this is an actual disease pathway, what kind of disease is this? And we have our categories of novel types of diseases of interest. So we also pin them like that.
Mm-hmm. Okay, that makes sense. There are more questions coming up about the time and effort you had to invest. So apparently lots of people are impressed by your knowledge graph. So there's a question from Louis. Which part was the most complex and time-consuming one? The screening, and I assume he needs data screening in this case, the curation, or another one?
If we were to do the tasks sequentially, I would say the curation process for the disease enrichment would be the longest one. But we do that in parallel. So there's this company that does the manual curation for us, and for doing the manual curation of the obesity, it plus the osteoarthritis in the context of obesity took around 5 months. Mm-hmm. And then for the implementation of the GDS pipelines for the target discovery, because those ones were modifications from public ones, they were not as time consuming. I would say, I don't know, they were 4 months each one, more or less, in parallel.
Okay, so people in the chat, if you have follow-up questions or this did not answer your question, just write a message. I will follow everything that we did. Perfect. There's another question from Laurent. He asks, you defined multiple features in the nodes. Is that in the graph database itself, or do you use another database and generate different knowledge graphs as subsets? So this is, I think, about your infrastructure. It's really everything in Neo4j. Do you have other databases, other data stores?
Yeah, that is a really good question. So with the GDS provided by Neo4j, you can't really have a subsetted knowledge graph that you work with on the fly. You need to create it again. So for example, for the Hethionet modified approach that we took, the part where we do the randomized graphs, we actually have to export it as a JSON file and then make a new one and then apply the randomization algorithm there and save it and do that several times. And then we delete all of the randomized ones and keep only the original one. So one thing to keep in mind is always have a good backup.
I think that's a very general advice, right? Perfect. Let me have a look at the chat if there are more questions. And again, to the people who ask your questions, if you have follow-ups, just write a message in the chat. I will see that. Since our next speaker will be a few minutes late, I have a question actually about the GDS library and the algorithms and all that. So just out of general curiosity, how much of the analysis can you do within Neo4j? So can you maybe describe a bit what other kinds of network analysis tools, data analysis tools, machine learning frameworks, for example, you have used outside of Neo4j and how you interact with them?
So, for example, for the novelty score, the DWPC was something that we implemented, but then of course we used the querying of Neo4j. And the other sort of ranking of the scores is within Python. So it's outside of the Neo4j framework. The ranking based on the closeness to the disease, the one where we use 3 different kinds of algorithms, that's all Neo4j. We calculate them with the GDS from Neo4j, and then we extract the numbers. And in Python, we do this little normalization and merging of the 4 different scores. And for MindWalk, it was all Python scripts.
So you use partly GDS, and then you interact with other data analysis frameworks.
Yeah.
And how well does that work for you? So how well does it work for you to extract data? Because then you need like bulk data, right? You have to you need big tables essentially for most of the data analysis frameworks.
Yeah, it works pretty well because, well, in these ones that we have applied so far, we use all of the graph. It is going to get a bit trickier when we decide not to work with all the graph. As we keep enriching it and we say we want to focus on the information that does not include the novel knowledge because, I don't know, say that is going to bias our identifications or something, then we will have to do some fishing or subsetting of the graph. And then, again, fish it out, put it into a JSON file, and make a new graph and work on it. So it's just, yeah, there's going to be that intermediate step.
Mm-hmm. Okay. So let me check the chat. Lots of things, impressive talk, impressive knowledge graph. But I don't see, there's another question. Christos asks, could you elaborate a bit more on the architecture of your systems? Also, and I think this is a super interesting question, do you update the graph and how regularly?
Really good questions. For the infrastructure, we actually need to make it more powerful because for the things that we are running is starting to the analysis take longer than we would like them to. We have it all set up in AWS instances. We have only 8 cores. and I think like around 60 gigas or 30 something gigabytes. So it's not as powerful, and that is actually one of the next planned steps to increase its computational power a bit more. And then regarding the update of it, that is also a tricky, good question, because we started working on it and kind of trying to see how it works and get the experience with it. The way we designed it was from scratch, and it was more or less an on the go. If we need to change it, we change it, and then we learn. So right now, we are not working with it as if it was a production product. It is an internal one that we decide whenever we are going to update it, because we pull from so many different sources, not all of them update at the same time. We would kind of do it as specific interventions that we defined. And we finally got it to, well, like you saw, right now it is a little bit of a nightmare. And right now what we're doing is polishing it. Once we get to the polished schema, then we actually have plans to update one of the data sources that I know they released a new version.
Mm-hmm. Okay, so I think this is really an interesting question, because the update is something that comes up a lot, because the knowledge that is out there changes, obviously.
Yeah.
So maybe one last question, and for the audience, so the next speaker is a bit delayed. We will either swap the next two sessions or pull another session from later a bit earlier. But there's room for one more question, maybe. Again, an interesting one. From Thomas, he asks, do you have any suggestions how to find mappings between various medical databases?
How to find mappings?
How to find mappings between medical databases.
I'm not entirely sure what you mean with find mappings, but it is, for example, the first thing that we got a hold on was gene ID mappings. That we know not all the IDs map one-to-one between the different data sources. So we did quite, we spent some time making sure that our mappings were correct for that. And then also we spent quite a bit of time as well mapping the disease IDs for the different ontologies. So for example, the human disease ontology, it doesn't cover all the diseases that we're interested in. So we had to complement it with the human phenotype ontology. And then maybe also that one didn't have some that we were more in-depth interested in. So we use the NCIT and so on. So once you start merging a lot of different ontologies, you need another ontology framework to support you better. And for that, we are using the Novo Ontology Department in Denmark to help us with.
Mm-hmm. Okay, let me check. So I think that was everything in terms of questions. We had a lot of questions, a lot of interesting ones, a lot of relevant ones about updates and so on. So I would say thank you, Lizella, for your fantastic talk, for taking the time to answer all the questions, and hopefully for sharing as much as you can in the publication.
Thank you.
So thank you so much. And the next speaker is late. Alex, should I continue maybe?
Yes, I think that probably makes sense. We have you as our joker, so you can basically go anytime. Hopefully, the next speaker will show up in the meantime, and then we can just continue. But yeah, I think that's a good idea.
Okay, perfect. Let me share my screen and show you the next talk. Perfect, you can see my screen, right? So the next talk is a bit delayed. So I'll simply continue. We're going to put the Health Echo presentation into this slot, and I hope we can simply swap the slots. Okay, so I continue and give a brief introduction of Health Echo and what Health Echo is all about and what we're going to do with Health Echo. So let me start this presentation. Perfect. Health Echo is all about bringing the world's health knowledge to research and medical decision makers. This is our claim. And what is most important about Health Echo is that it's an open source, non-profit collaboration. So it's an open community with a goal to build knowledge graphs for medical research and for healthcare. Health Echo started with the COVID graph project in March 2020. So a couple of people working with knowledge graphs, working with Neo4j, found data sets around COVID-19. And apparently that was a very, very interesting topic and very important topic. And we just started by building a knowledge graph, by collecting data and putting it into a knowledge graph. And to be honest, the main reason why we did that is because this is what we do. So this was a hobby project, a fun project, a side project. But then it really grew. So we started posting about it, talking about it, giving presentations, and a lot of people joined our community. So in total, we had contributions from over 30 developers and lots of interesting contacts in our network. But it started with COVID graph last year. Health Echo is a collaboration, and I think this is very interesting, very important, of public sector and private sector organizations. So we are small companies, big companies, and public research from various universities around the world that support the project. And I think this is really interesting. And what I really like most about Health Echo and about the community is that we are still open. So everyone contributed something, all the code is open source, the database is open source, and various tools to access the data are also available for free. So I really, really like how this community grew. And is still very, very active. This kind of summarizes our mission. When you collect data, you have to do something with the data, right? You want to create some sort of effect, some impact in the end. And the question for every knowledge graph is, you have all the data combined, that's great. You can run queries, you can do data analysis, but what do you do with it? And we essentially see three ways how we can bring our data to the patient in the end and improve the patient's life. The first one, and this should be obvious after the last talk, is to work with researchers. So a lot of the data that we have in COVID graph is very closely related to the data that we saw in the last talk. So working with researchers and building, knowledge graphs that everyone can use can be very impactful. On the bottom, we have the very other end of the spectrum. We had very interesting discussions with frontline health workers on tasks like working with treatment guidelines. As a medical practitioner, you get treatment guidelines. You don't have the time and the resources to investigate all the research behind this. And putting knowledge together and making it more accessible can be incredibly valuable for the people who are actually treating patients. And a last branch, essentially, and one that really just came up over the course of the project is that we can essentially provide data to policy and decision makers. And I mean, during the COVID lockdowns, we've all seen and witnessed firsthand how difficult it is to take the right decisions. And making data available and putting it in the hand of the people who take the decisions in the end can maybe have the biggest impact in the end on patients. We all know this problem, right? We are researchers, we are knowledge workers, and we all know we can't never keep up with the stuff that is published. And in the very first data set that we loaded into COVID graph, we had, I think, 40,000 publications. That was a curated publication data set from Microsoft Research. And this grew to 150K over the last couple of months. And obviously, no one can read all that. And linking the information that we find in these publications is something that is very, very difficult, but at the same time, obviously, very necessary. So here's just a simple example. We have gene-related data on the right side, protein-related data on the left side. And even combining protein people with gene people, protein modification people with people talking about gene variants is very, very difficult. Because what you need in the end are all these links. And I think this is related to the question Thomas asked after the last talk. So how do you combine, how do you find mappings between all these medical databases? And this is a significant part of COVID graph. So gene and protein data is obviously linked. We use databases like UniProt and NCI-Gene and Ensemble and so on. But in the end, the difficulty is we don't just have two databases, we have so many different ones. And for every kind of relationship like between protein and gene, we don't just have one data source, we have multiple data sources. Here's a brief overview of the stuff that we have in COVID graph already. We have pathway data, we have a huge focus on publications. We have much more than that. We don't just have the curated publication data set, we also have patents. I think that is a very interesting data source and definitely an underused data source in the biomedical research world. Here's an example of the gene and protein work that I just mentioned. So the basic biomedical entities. What we can do is we can use these biomedical entities to link text. We load a curated patent and publication data sets with a text annotation to link everything. And then you can ask questions like, how are my publications, maybe you have one publication talking about proteins, another one talking about genes, but how are they linked? This is just a rough overview or estimate of our data sources. We have roughly 30K patents, that's in blue in the top left, 150K publications. We have clinical trials, we obviously also have statistical data, and then we have basic biomedical data and annotations like pathways and ontologies. We have a lot of plans for the future, so COVID graph is kind of the inaugural project of Health Echo. But our goal for the future is to go beyond COVID and release a second iteration of our graph next year that will go beyond these curated or COVID-focused data sets and expand it to the full PubMed, for example, and the full clinicaltrials.com. We do a lot of data analysis. Like NLP, for example, where we extract data, where we extract entities from text to put it into our knowledge graph. Our data is available, and I think this is very, very important. So everything that Health Echo does has to be open source, free, and accessible. So there will be no closed source applications here. You can always go to covidgraph.org or healthecho.org. You will find all the links there. We have a public SAMSPECT installation. SAMSPECT is a super interesting graph exploration tool that works on a slightly higher level. We have a public Bloom. We have our own custom applications as well. So lots of interesting ways how to access the graph. And well, obviously the Neo4j browser is here, so you can access the graph directly. We worked on very interesting use cases all the time. So a couple of things are just very obvious, right? If you have a lot of genes which are linked to publications, you can think about rankings, and you can use graph algorithms like the PageRank to identify the most relevant genes. And this should not be a surprise. The ACE2 receptor is actually the most important gene in a COVID-19 focused publication dataset. And we all know ACE2 is the cell surface receptor that the cell uses, that the virus uses to get into the cell. So this is obviously an important gene here. We started over time, a lot of collaborations with researchers and existing Neo4j graphs. So there's in the systems biology world, so when we actually do systems modeling of biological mechanisms, there's a database called RASIMOS that collects these data, these models, these SBML models, and this was very interesting because it was an existing Neo4j graph, a Neo4j tool and project, and it was super easy to integrate this. And what we can do now is we can go from this ACE2 gene, which we identified as being very important in this context, down to mechanistical models that already exist and show us how this gene works. So this was a very interesting collaboration. the pattern data set, I think, is something that is for sure underused and can, give really, really interesting insights if you learn what people work on, for example. So this is a use case where this SAMSPEC data exploration tool can be very handy. It's a tool where you can define higher level meta paths, and then we can filter on every step. And we use this to investigate which companies are working on what kind of genes, and we work with that. And another thing that we did here is to work with researchers in Greifswald in Germany to search for a specific gene and understand how this gene is annotated, and see the papers that are published about this gene. It's a gene that was in clinical research somehow implicated in COVID-19 patients, and getting a first understanding. You don't really know what you're looking for. The goal is to get a first understanding of the knowledge landscape around a particular gene. And for this, a knowledge graph is obviously super powerful. And this is just a zoom into this graph exploration. You can see here that we have genes which are related to other genes, and then we go down into the branches of pathways, publications, and also these systems biology models. We had a very interesting collaboration with a company called Meltwater. They provided us with a sentiment analysis data set of press releases from pharmaceutical companies about drugs. So that's an interesting concept, I think, because a lot of information becomes public before it is, published in a scientific report or paper. And we had a couple of interesting examples where we looked at the sentiment, positive, neutral, negative, for the various drugs that people talked about. So remdesivir looks green, hydroxychloroquine not so much. Health Echo is all about collaboration. And we are a growing community. As I said, we had active contributions from around 35 developers. And in total, I think 150 or so people who joined our community at some point. And with Health Echo, we do have goals for the future. And one of the goals is really to publish a second iteration. Our internal working name is Everything Graph. We want to go from a curated data set of publications to full PubMed. We want to go for the full clinical trials databases. We want to attach to larger patent databases to really bring it to the next level, to not just focus on COVID, but make it available as a resource for disease research in general. Another thing that is, and I said that like 500 times maybe, but it's just very important, we want to stay open source and completely free. And I think this is very important. And we always want to stay open for researchers from public sector, but also for small companies and for pharma companies, as long as everything stays open source. And what I can kind of envision for Health Echo is that it could develop or grow into the place where we all come together. And a couple of, I would say, more specific examples are sharing of data models. So we all work with genes and with proteins, and obviously the protein information comes from UniProt. And we all get our protein information from UniProt. Would it not make sense to do that, if we all do that in the same way? Sharing data models and sharing, data paths, let's say data extraction pipelines. I think that would be really, really useful. No one loses, everyone benefits. And I think Health Echo could be the place where all of this comes together. So I can just invite you, go to healthecho.org. We're very active on LinkedIn, so we can follow our latest updates, what happens at Health Echo. And we're definitely always open for contributions. And again, all the data is free, knowledge graph is accessible, and with various tools that you can use to explore the data. Okay, so with that, I think I'm done with COVID Graph at Health Echo. And if you have any questions, just shoot. Maybe I can look for my own questions in the chat. Or Alex, you can do that maybe.
I can definitely do that. And we have, I think, one question, which we have time for maybe. Ibrahim asks, how do you extract the information from unstructured publications?
And that's a very interesting question, because it's kind of the heart of COVID Graph. It's something we get a lot. And one of the reasons why I'm super excited about the presentation about the Lifelike platform later and the NLP project tomorrow, because this is the most difficult aspect. I mean, in a certain way, NLP seems to become a commodity these days, right? But if you look at it from a more practical perspective, it's not that simple. So we tried various model-based NER methods, BERT-based, for example. But the bulk of our data is what Lisandra described as text mining. So we don't have fancy models that do that identify entities we search, with obvious limitations of this. And just as a call to the community, this would definitely be one of the areas where we could need support. And where we could really need, someone with more NLP experience to, maybe expand this a bit.
Okay, cool. Thanks, Martin. Maybe we can squeeze one more in before we continue with Federico. In our track is from Angel. Minor but important issue, and I think he's right, is how did you clean and pre-process the raw data? Similar to what you just said now is how do we make sure that the data you load in is in the right format? How does it fit and make sense with all the rest of the existing data?
Super obvious question. Everyone asks it. We don't. I mean, to some extent, we have to trust data sources, right? So we download proteins from UniProt, for example. And the approach that we take, and the approach that I think can actually make a lot of sense, is you cannot be better. You cannot be better at protein research than UniProt, for example. And I think the idea is to be very transparent about the data that we have. I mean, if you work with R, for example, and you use R packages for data annotation and ID mapping and so on, you don't see what happens, right? Maybe you can get from a protein to a gene. You can say that a certain variant is associated with disease, but you usually don't know what happens. And our approach for COBIGRAPH is we at least make it transparent. We cannot solve the kind of inherent difficulties and problems in the data. What we can do is we can make it transparent. You always know if you go from one data set via genes and proteins to publication, for example, you can see the path. And on every step, you know where it comes from. And step by step, you can create paths and ways to navigate this network that you think are reliable.
Cool. Super. Thank you very much, Martin. Thank you for swapping in. I think next up we have Federico, but I guess you take on introducing our next speaker. So I'll let's swap.
Yes. So it's a pleasure to announce Federico, who's been very active in the Neo4j community for a long time. And he's going to show how LifeGen, his company, is building complex web applications for healthcare using Neo4j. So the stage is yours, Federico.
Thank you, Martin. Can you hear me okay?
Yes.
Perfect. Then let me just try and get my slides here in the same screen as I have this. Okay. And try and share. And okay, good. Just gonna check if you can see my slides. Can you see them?
Yes, you could go full screen maybe.
Can you see them now?
Yes.
Okay, perfect. And thank you for the invite. Yes, so a quick introduction, Frederic Braga. I'm the Chief Technology Officer at LifeGen. Before that, I spent a few years with IQVIA, and before that, for the last 11 years in the life sciences industry. I have a background in software engineering and management, and I've done a few things with Neo4j. I started working with Neo4j to solve some of the management in life sciences. The first time I came across, going back in history, it was one of the first problems I used where I used Neo4j to solve for that problem was in the clinical data warehousing space, where you're trying to put together clinical data from sponsors to generate submission data sets. And there's an underlying issue with matching data, because you're collecting a bunch of transactional data around study data, for example, that needs to be matched with some master data and some metadata, but that metadata and master data itself is evolving while the study is ongoing. And so I needed a tool that would allow me to relate data a bit more flexibly. Initially, went from SQL to some of the NoSQL databases, but then you hit all these problems with performance around joining queries, joining data, which document databases, most of them are not really made for, and started using Neo4j. And the topic I bring for this talk is a little bit of, I think, just a little bit of division of that history, because when I started working with Neo4j, I still remember that in some of these presentations, people would say, well, Neo4j is great, and some of even the Neo4j engineers would say Neo4j is great, but it's not the tool for all jobs. This kind of, maybe you want to use SQL databases if you want to do something. Of course, there is no silver bullet for everything, but practicality sometimes is more important than perfection in computer science. And so we try to, what I bring for this talk is really why and how we decided to use Neo4j to be our core database for our entire application. And we'll use it for basically everything. We try to simplify and normalize on that stack. And so before that, I'm just going to give a quick introduction to LifeGen, company I work for. So LifeGen is a software as a service company. Our mission and vision is to help the development of value and outcomes-based health care. So why is that important? Because we believe that value-based healthcare can help more patients to receive the healthcare treatments that they need. The history of LifeGen actually, so I'm just going to go a bit over the introduction, and then I'm going to talk and explain a little bit what is value and based agreements, why we built the platform that we built around Neo4j, the style of our platforms and learning, share also a little bit of what's next. So we're, just to mention, we're considered one of the top 10 startups last year, certified, a member of InnoSwiss, Swiss-based company, but now more and more with more international customers. The story of LifeGen is a story of a company that started, and if I can play this video here, I don't think so, it's on our website. It's a story of a company that started with Sophia. She was a 3-month girl, which was unfortunately diagnosed with SMA, spinal muscular atrophy, which only gave her 3 years to live. Despite being based and living in a rich country like Switzerland, the healthcare system did not cover for the 1.2 million Swiss franc treatment that Sophia needed in order to have a chance of surviving. This therapy existed, but the reason why the healthcare system does not cover for that treatment is an interesting one that leads to the whole topic of value and outcomes-based healthcare, and the sustainability of the system that we have in place, not just in Switzerland, but in many, many countries, the healthcare system. Why is that important? Because without the right incentives in place, we all know that without the right incentives, financial incentives, there is no innovation, right? Many of us would not be standing here without the existing financial systems that are in place. So what we aim to do is to correct this system, to give the main stakeholders the tools to correct the system, and to for them to establish agreements where the reimbursement or the payment for the treatment is based on its value or outcome and less on its volume. And that means on a patient-by-patient basis. So that is our mission. Next slide. And what we needed to do this essentially is we needed to build a platform that would enable both stakeholders, essentially the payers and the manufacturers, but also actually sometimes the providers of the healthcare providers, hospitals, to connect under the same platform, to establish an agreement, to have all of these agreements in one platform, to see real-time insights, to measure the progress and the performance of these agreements, to automate and simplify the whole and remove the burden of collecting data, to enforce it, to do that in a transparent manner and with the highest level of security and access controls. This platform also needed to be scalable. So the reason of scalability is that you want to have the same platform because it is a network platform. Let's say a certain payer, for example, a healthcare system in the United States, they have multiple agreements with multiple manufacturers, and these manufacturers per se in the same country have agreements with other payers, and therefore they can't all be using different versions of the system or even different systems. That would be extremely inconvenient. So in order to facilitate that scalability, we have to build through software as a service, single instance, multi-tenant application. This application or this platform had to be modular. So we have a number of modules from models. So these are template models that payers and manufacturers can choose from and select, well, for this agreement, this is the model, the financial model or the outcomes model that is more suitable to what I see is fair for the execution of that agreement. And they have another module where they have their agreements, can visualize a lot of the performance of those agreements. Then there's other modules like cases where a lot of the financial management happens, as well as the financials, which is really more on the operational finance part, and a data module where we ingest and manually also give the opportunity also to manually enter data to the users. So it's a data-driven platform that is flexible and scalable. It's independent and trusted because we're not catering in interest. It's an important point. Both the providers and the manufacturers trust us because they're both our customers. We want to serve them as best as we can to all of them, and it's compliant and secure. So I think I've touched a lot of bullet points here. The next slide. Just a little bit of the stack that we use. Very simply, this is simplification. We have a client application. We have a number of, let's say, proxies and security layers in the middle. We have a number of proprietary services that we built in order to support multi-tenancy, to support GDPR, to support anonymization, and data privacy. More or less in sync with GDPR. Then we have our APIs, and we went for what's called a GraphQL first approach of our APIs. So all our APIs are based on this standard called GraphQL. And in the backend, we have a graph database, which is Neo4j. So why did we choose Neo4j, and why did we only choose Neo4j and didn't decide to do a number of other things? Well, first of all, because we know that we want to leverage the data that we platform to conduct a lot of analysis. So, if we database, for example, just to support the application, then somehow ingested that data into a graph, then you get into this normalization, denormalization. You spend a lot of time and effort normalizing and denormalizing your data. Second of all, if we decided to go for some sort of document database for indexing and fast performance, then we would lose a lot of performance because we have a very complex data model with a lot of relationships and a lot of entities. And so here on the right, you just see a little snapshot from our GraphQL schema, and we have around 804 objects, which is almost of the entities that we have in our data model. So as you can see, not really a simple application from that standpoint. And all of this data is connected and it's complex. So so these are some of the reasons we went for Neo4j. Also, we wanted to implement this we wanted to use GraphQL because it's extremely in sync with this FEF software development pattern that stands backend for frontend. So essentially, it's a trend where you're essentially building your backend and thinking essentially about your front end when you build your backend, and make sure that that you build your backend in a way that it's friendly to then build a front end for the data to be queried, for the types to be aligned, for the data models to be aligned, and essentially to give a lot more flexibility and speed to the application development, the front end developers. The fifth reason we chose Neo4j is because we love Cypher. It's way shorter and faster than writing SQL statements. And really, I think the GraphQL approach I already mentioned, but at the end, I think this is the overarching argument. We saw that we could develop our application much faster using GraphQL, and that Neo4j, with the way that it essentially compiles a GraphQL query, was the most performant approach. So many of you probably don't know this. Essentially, the way GraphQL works is that it creates a type system that stands somewhere in between your front-end application and your database, and that enforces some types to the data flows, and at the same time creates a data graph, right, with these types. So essentially, what it does is it resolves these. It knows that if you have patients and treatments, and that they have a relationship, it knows that if you query a patient, and then if you want to see the treatment that is related to that patient, it has these two resolvers, patients and treatments. And if you query the treatments from the patient, you can query. This creates what is known as the N+1 problem. You're generating sometimes an exponential number of queries for your data, but the way that Neo4j handles that is very gracious, essentially using one of the epoch functions, but essentially the power is in the expressions, where the return of the statement is essentially an expression of that query. And so the performance is amazing for GraphQL. It's very simple to implement. And there's this package called the GrandStack, which we really like to develop. And so we went for that approach. Okay. So some of the reasons I had some doubts about Neo4j is we were using some .NET infrastructure, and we had some problems with the driver between .NET and Neo4j. We thought about a little bit of vendor lock-in, of course. That's always there. And we started to see, this is a little bit later on, actually, some scalability topics where the complexity of how Neo4j handles clusters is a little bit left to the developers. That has to do with how atomic transactions are managed in a cluster, and let's say the methods that are implemented at the Neo4j driver level to handle the transactions in the cluster mode. So you end up having to write more code than we would like to when you have to do that. But I would say, so in the last year we developed our platform. We have more than 150 now running value and outcomes basic units running in our platform. We went live 6 months ago and started bringing our first customers into the platform. And the speed of development is just incredible that we're able to achieve, I believe, with this stack. So we're very happy with that decision. Some of the pitfalls and learnings that I can share. So we went with this with the GrandStack, so the GraphQL stack that was available as part of the community, yeah, that was no longer supported by Neo4j. Neo4j is now officially supporting a new GraphQL package, which we, in general, very much like. But unfortunately, we started using the old one just, I think, 5-6 months before it was no longer supported. So actually, we spent the last 2 weeks migrating our entire application from the old package to the new package. So we have to do that. There was some missing functionality in the old, in the new package, which we, if someone is using GrandStack and thinking about migrating to the new package, I'm happy to share our experience in more detail. And but we definitely see the benefits of using the new package and see the standardization of how queries are managed against the GraphQL standards, with less special cases on how the selection sets are exposed. Second of all, some of the learnings we had, as I mentioned before, is around the handling of the cluster. So we have had a few issues when we started then scaling into a larger cluster and concurrency. And one of the issues also is that if you're trying to query through a session or a transaction that you run, and then you try to pass or read some data in the middle of that function with a GraphQL query that you fire through, for example, the OGM that is in the new package, or that's very difficult to handle and requires a lot of code refactoring. and close monitoring, because it can introduce performance issues, issues with your cluster. And yeah, so we're doing a little bit of code refactoring and implementing some best practices within the team of how we handle these things. Yeah, also happy to share those learnings if someone's interested in more detail, more technical detail. And the third one is we're a small team. We're including myself, 5 developers, but we have to we use Azure, and there's no option to, as far as I know, to have like Aura on Azure, like you have for other databases, like MongoDB, for example, or SQL. Azure has services that support these databases. That does not exist for Neo4j, so we had to stand up our own virtual machines and build a cluster and maintain it. That's really not very, very nice. It's not what we thought to do. So we're actually considering Neo4j Aura, and we very much welcome that offering. And I think, yeah, the final point is what's next. So we started, we're initiating, we have plans to very soon have customers in our platform in other geographies that require us to have our data in there. So we might have to use the federation functionality of Neo4j and then scale the cluster outside. Horizontal scaling is also an issue with both on the GraphQL level and then in Neo4j. And we need to say make our infrastructure a little bit more robust to cater for potentially thousands of users. And we are also in the team one data scientist that is really usage of the out-of-the-box Neo4j algorithms. For example, some of the data that we have to infer real-world evidence data. So as we, in many cases, have the insurance companies upload a lot of the medical claims, and the insurance claims, we can see, let's say, somehow infer the efficacy of a treatment in certain contexts. So using the connected data that we have in the graph and leverage algorithms will in the future allow us to infer true efficacy of treatment, which is what a lot of the academia and other stakeholders are interested as well. But that's more a little bit into the future, and we once we get a little bit more scale. We already have a lot of data. We all know those things normally require large amounts of data. That was my last slide. I don't know if there's any questions, Martin. I'm just going to pause there.
Yes, so thank you for your talk and for your presentation. I think your pitfalls and learnings are something that can be very interesting for a lot of people here. So especially working with a cluster and the details here, that's something that I think a lot of people can find very interesting. So we have a couple of questions. Let's start with the first one. So there's a question. Hi, Federico. Is medical equipment and external service providers also included?
I'm sorry, I didn't hear. Medical equipment. Was that the question?
So is medical equipment and external service providers also included in the SIMT?
Yes, so we have customers actually that are both medical device and drug manufacturers. So, and we also have hospitals that are logging into the platform. Actually, just today, one of our customers was presenting it with a use case here in Switzerland in cardiac ablation. customer is a public one, it's Johnson & Johnson, where for their innovative agreements for some of the devices that they launched for cardiac ablation, which is a very common surgery, right? They implemented value and outcomes-based agreements. So the price that the hospital pays for the device is related with the need for a second intervention, for example, which they claim that with the device is not necessary. So essentially, this is a way to allow the pharmaceutical manufacturers and medical device manufacturers also to differentiate themselves, right? Because fortunately, in a world where there's competition, with a system with value-based agreements in place, the one that benefits at the end is the patient. Right? So there's more access to the treatment, and there's a price for the treatment. So yes, we support
that. Okay, and there's a follow-up question from the same person. How are indirect stakeholders like community services included?
So far, like we don't have. So far, no. We have the possibility, let's say, built into our platform, to have multiple stakeholders that can see the agreement and they can see different things. So we have essentially the payer, the buyer, and we can have observers into the agreement. So it is another entity, but essentially the main party parties, but it can be 3, 4, 5 end parties can have access to the agreement as long as they are contractually in agreement to allow for that, to log into our platform and see the performance of the agreement. And we've implemented services around data privacy, where we can configure every field in the platform to be hidden or visible, depend for each entity or each tenant of that for that specific agreement. So on an agreement basis, one party might be seeing some data, the other party might be not seeing that data. So it's just hidden or masked, and other parties might be not having access to that at all. So.
Okay, so unfortunately time is up and the next speaker is waiting already. So Federico, thank you so much for presenting the work, presenting LiveGen, and sharing the pitfalls and also the GraphQL insights. It's super interesting, I think. So thank you so much.
Thank you for the invite, Martin.
And we can now continue with the next talk. Jérémy Grignard, data scientist at Servier in France, will present Pegasus, a knowledge graph to support drug discovery. So Jérémy, stage is yours. Really looking forward to your talk.
Yes, Martin, is it good? You can hear me clearly?
Yes.
Okay, cool. So thank you. So yes, as Martin says, today I'm presenting you Pegasus, which is a knowledge graph to support early drug discovery at Servier. So my name is Jérémy Grignard. I'm a PhD student preparing thesis at École Polytechnique and working in the Servier Research Institute, as well as INRIA Saclay, under the supervision of Thierry Dorval and François Fage. So for the presentation today, we'll give you some industrial context about our different activities. After, I will present you in more details the Pegasus data model. And after, I'm going through different early drug discovery applications we are currently using at Servier that use Pegasus. So for the industrial context, so we are working in the drug discovery domain, and drug discovery process is a very long, expensive, and very risky process. And basically, many failures occur at different stages in the research phases, in the development phases, and particularly when failures occur in phase 2, it's really critical for the company. And it's partially due to the fact that the hypothesis that links the disease and the target is really tested under real pathological conditions. So with my team at Servier, we are working in the early drug discovery pipeline for target identifications, target validations, improving screening campaign, as well as de-risking experimental and new experimental modalities by using different technologies and computational methods, such as phenotypic profiling, such as machine learning, sequence designs, system biology, and of course, knowledge graph, that is Pegasus I'm presenting you just after. So why Pegasus? The rationale of Pegasus. So basically, you all know that data in the pharmaceutical domain are really heterogeneous. We can have experimental data, we can have predicted data, and these data are coming from multiple sources that are siloed and lack contextualization. So we can have proprietary data, collaborative data, consortium data, commercial data, and the well-known public data. You just described all of you before. So the objective of Pegasus basically is to support the rationally the decision-making at Servier by capitalizing on all already available data in vitro, in vivo, and also by accelerating the development of innovative applications to support our therapeutic projects. So about Pegasus data sources. So as I previously mentioned, so we have public data, commercial data, consortium data, collaborative data, proprietary data that are fed into the knowledge graph Pegasus. So let's take a couple of examples about public data. We have NCBI, Ensemble, Reactome, GEPTEX, Human Protein Atlas, and so on, the well-known available public data. Commercial data are basically data we can buy. Consortium data are international consortium where Servier is involved, such as JumpCP consortium and the IMI-UB Open projects, what are two consortiums that aims to identify for the IMI-UB Open projects chemical probes to modulate particularly and actively single target. And the consortium JumpCP aims to perform at an international scale high content screening experiments with different pharmaceutical partners. And of course, proprietary and collaboration data are data we can produce internally. So Pegasus first is based on all this data, and Pegasus is also based on our capabilities, internal capabilities and expertise, so such as the phenotypic profiling. So phenotypic profiling is basically, the objective is to identify perturbators such as chemical, antibody, microRNA, and so on, that can modulate a target of interest. And when we are doing high content screening, high content imaging screening, instead of measuring one single variable such as the activity of the target, we measure the phenotypic state at a single cell level of different cellular model of study. And within Pegasus, because we have plenty of experiments of phenotypic profiling experiments at Servier, we take advantage of this data by integrating into Pegasus phenotypic profile similarity measurement. So this is for an example for phenotypic profiling. So for in silico machine learning algorithm, we also integrate into Pegasus chemical profile that measure chemical similarity between two compounds. For instance, a compound A can be similar to compound B according to a Tanimoto algorithms, according to a cosine algorithm, according to a DICE algorithm, and so on. We can do the same thing for sequence profiles. So for instance, by homology, microRNA can be homologed with a given percentage to another microRNA. And after, we have different analyses more about functional profiles. So basically, one gene or a set of genes can be functionally related to a set of other genes, for instance. So this is a big picture of Pegasus data sources. But you can say, okay, why developing a new knowledge graph while there are plenty of knowledge representations in the literature? So basically, we have two ways of representing knowledge using the well-known ontologies, such as gene ontologies, bioassay ontologies, CMPO ontologies, some disease ontologies, and so on. And we have really knowledge graph, which are, when we use Neo4j technology, they are label property graphs. So here you have a little example of some knowledge graph that are used in the drug discovery pipeline. So it's come from a paper which is not yet published, but it's on bioRxiv or arXiv, where you can see different knowledge graphs that are used mainly for drug repurposing in the pharmaceutical domain. So why Pegasus? Because all these graphs are not suitable for our drug discovery applications, because they are not really the notion of transcripts. Generally, in the cited graph, you have a gene that is representing also a transcript that is also representing a protein in one single node. And there are also no notion of chemical, biological, and phenotypic similarities. And there are not several chemical and biological perturbators, such as antisense oligonucleotide, PROTAC, and so on, that are present. So we decided to create Pegasus. So here you will see just right now the high-level Pegasus data model. So we have five main entities, main nodes. So we have biological entities, high-level entities, sequence structure entities, activity entities, and perturbators entities. So biological entities, we have a notion of genes that can be transcripted into several transcripts. Transcript can be translated into several proteins if the transcripts are protein coding, basically. For the perturbator, we have different classes of perturbator integrated into Pegasus. We have a small molecule, antibody, and all therapeutic RNA when we are interested on. Here, each biological entities and each perturbator entities can have sequences or chemical structures. So let's take an example. A small molecule can have a chemical structure. A protein can have a sequence or a 3D structures. And based on the structure itself, we can compute similarity measurements. So let's take a drug that is chemically similar to a Servier compound, for instance. After, we have high-level objects in purple. So it's the name indicates what are these kind of objects that are really high level. We can have the notion of study models. So let's say tissues, cellular model of study when we are performing screening campaigns, for instance. We have the well-known notion of disease, of biological processes, molecular function, cellular component, and of course, phenoprint. A phenoprint is a phenotypic signature that are induced by a chemical during a screening campaign. And the central notion of our data model is a node activity in yellow. So a node activity is basically, the aim of an activity node is to contextualize the relationship between two objects. Let's take an example. A small molecule has an activity on a given protein. And this activity has been measured in a particular tissue of interest or in a particular cellular model, and so on. So really, the activity node is to give context about a relation between two objects, because a drug A can activate or inhibit a protein B, but in a given context, and so on. So this is our high-level Pegasus data model. There are a couple of notions that are not present to make it easy, quite easy to read for the presentation today. And really, a main point is that our data model reflects our therapeutic questions and projects we want to solve with a data model, because you know that data model, when you are doing graph, are really flexible, and our data model, considering different kind of questions, can easily evolve. And the last release of Pegasus, so the release of November, has about 80 million of nodes of 54 types, has about 100 million of relationships of 13 types. And the generation from the raw data is about 15 minutes to generate. And we are using a Python framework, which is named Kedro. Okay, so now I'm giving you and I'm displaying you some illustration of early drug discovery applications using Pegasus, and keep in mind, we are really therapeutic project oriented. So first, we have a use case that I'm using to Pegasus in order to design a focused chemical library for screening campaign. So we can ask the following questions to Pegasus, because he's really kind, basically. We can say, okay, Pegasus, can you indicate all chemicals that modulate a target directly or indirectly through target expansion? And because Pegasus is really kind, he can give us an answer. So for all results that will be displayed during the presentations, the results are not exhaustive for clarity, of course. So here on the slide, what you can see is in green, a target of interest. That can be a gene, a transcript, or a protein, whatever. And what you can see in red, it's different kind of chemicals, such as the drugs that have an activity on the target, molecules that have an activity on the target, a chemical probe that have an activity on the target, or a PROTAC that have an activity on the target. So here, each of our node and relationship have plenty of meta information, such as the activity node between a PROTAC and the target. We can set different information, such as the IC50, where the activity has been measured, and so on. One thing really interesting when we are using Pegasus is about chemical profile. So here, what you can see is Servier compounds that have been introduced into Pegasus with a notion of similarity measurement between a Servier compound and a molecule that have an activity of interest on a particular target of interest for a therapeutic project. The assumption behind that is that a similar compound should act similarly on targets, and it's a way we can select rationally a sub-part of the Servier compound library in order to perform really focused screening campaign. We have a target. You know that a target can interact with other targets, and through what we call target expansion. So let's take, we have our target of interest. A target can interact with another target with the well-known protein-protein interaction network, could functionally interact with a target because they belong to the same pathway and so on. And we can do the same thing. So if we want to modulate a target of interest, we could modulate one of his and one of its modulator. So let's say we could activate its activator or inhibit its activator and so on. So here, using similarities, we aim to select chemicals for its identification when we are performing a screening campaign just after the selection. Another real example is about repurposing perturbators using phenotypic signatures. So we can ask Pegasus, okay, Pegasus, can you indicate me similar perturbators that induce similar phenotypic signature? So this is one result that can be given by Pegasus. In red, what you can see is different kind of chemicals, such as Servier compound, molecules, Servier compound, that have an activity in a particular context, but here, the compound induced a particular phenotype. So if we take this compound, imagine that this is this one. So this one will have an activity and induce this specific phenoprint. This molecule on the right side induce this specific phenoprint. And what is the link between the two compounds here? It's a chemical profile. And what is the link between these two high-content screening profile is the similarity between the two phenoprints. And here what is really interesting is we use phenotypic similarities to link the chemical world to the phenotypic world in an innovative way in order to perform plenty of data analysis after. And one thing really interesting here also is it will allow us to jump to one experimental modality to another. So let's take this example. This chemical induce this phenotype. This chemical induce this phenotype. These two phenotypes are similar. And we could imagine that we have other experimental assays where we tested silencing RNA, microRNA, oligonucleotide antisense, other kind of perturbators that induced this particular phenotypic signature or phenoprints that are similar to them. So it's a cool way to jump from one experimental modalities to another. One thing really interesting also with the phenotypic signatures is about using an ontology that describe high content screening experiments. So here what you can see is about silencing RNAs that have an activity in a particular context. They induce a particular phenotypic signature. And this activity and this phenotypic signature is described with a CMPO ontology term. So on the left here, you can see a high-level example of what is CMPO ontology. So here what we ask to Pegasus is about, do we have silencing RNA that induce apoptosis phenotype using the CMPO ontology that are describing with controlled vocabulary terms the results of a high-content screening experiment. We have some project about trying to annotate automatically our high-content screening experiments. So let's take an example. So this high-content screening experiment, we can observe that we have apoptotic cell. In this one, we can observe that chromosome have an increased number of segregations. In this particular high-content screening assay, we observe that we have an increased number of cells and so on. So here we are using phenotypic ontologies to describe phenotypic results with controlled vocabulary terms. And that will allow us also to capitalize on already phenotypic results. So imagine that Pegasus is still present in 2000 years. And I hope so, as well as Servier, but not for myself because I will be very old. But we could use this kind of ontologies to ask, okay, in the past, what are the previous perturbators that induce a specific phenotype 10 years ago within an experimental and a specific experimental assay, for instance? Okay. Another example is about characterizing different kinds of perturbators, such as chemical and biologic. So here we ask Pegasus, okay, can you indicate perturbators that modulate targets that have essential gene activities? So what you can see in red here is different kinds of perturbators, such as ASO, microRNA, chemical docking. It's predicted chemicals that are used with virtual screening and docking and so on. So each of these perturbators can have validated and or predicted activity on a particular target. And here the target is a transcript. And because we have cross-references between all biological identifiers and references, so for instance, between NCBI, between Ensembl database, between Ensembl database, UniProt database, and so on, we can go back from the transcript to the gene and detect if the gene have an activity, is an essential gene. So an essential gene is basically a gene which is really important for life, and when it's deregulated, can induce particular disease or can inhibit or dysregulate some development and important development phases. So here we can characterize with Pegasus the effect of validated and predicted perturbators. And one thing really interesting, it's about when we are designing a sequence. So basically, we are designing plenty of oligonucleotide antisense for different kind of targets. And we have a particular project for a baby with a rare disease with a mutation on a particular gene. And we are designing ASO in order to target this dysregulated gene. But when we are designing this kind of ASO, this ASO have plenty of off targets. And here, basically, we are using Pegasus to characterize off targets of the ASO by minimizing the number of ASOs that can be that can target, for instance, genes that have essential activity, and so on. Really quickly about the ASO strategy. So we are using also ASO to target an interesting part of a transcript. So here we use Pegasus to de-risk an experimental modalities. So here we use ASOs that are designed to target UORF. So UORF is a upstream open reading frame. So it's in the 5' UTR part of a transcript. And our objective is to design ASO that will inhibit some part of the UORF in order to increase downregulated protein in particular disease. So here we ask simply Pegasus, okay, Pegasus, can you indicate all targets participating to a particular biological processes? So here it's, for instance, microautophagy. So here we have genes that participate to a biological process of interest. This gene can have transcript. This transcript have a UORF sequence, and we design some algorithms that can identify all UORF sequences for the human genomes. And we also have validated data about validated UORF. So this is a way of de-risking some experimental modalities, and we are planning to do some experimental validation on it. Maybe a global overview of Pegasus. So on the left side, you can see a results of Pegasus for rational selection of experimental modalities to support target identification and validations. So imagine we have a target of interest that is dysregulating in a particular disease. And we just ask Pegasus, okay, what are all the perturbators you can give me that could modulate this target of interest or that really modulate this target of interest? So with our current data model, we can so integrate ASO, BlockMIR, microRNA that will have a target, an activity on the transcript of a target. We have the notion of drug, of molecule, of proprietary molecule, so Servier compound basically, that could have an activity directly on the target. And by using target expansion, we can also say, okay, what are the different perturbators that can modulate target partners, basically? And here we can have UORF. With ASO, we can have chemical probe, PROTAC, antibody, and so on. So this is Pegasus. And maybe to conclude on how we really use Pegasus. So here you have Pegasus that help us to support target identification and validation for different projects, therapeutic projects. We use different similarities in silico artificial intelligence algorithms in order to add new dimension to the current data model. This can be done before generating Pegasus, or that can be done also when we have a current stable Pegasus versions, and do it on the fly within Pegasus. So it's really cool to make some prediction and so on, because we are geek, we like AI for life science and so on. But what really matters, it's about biological experiments that can validate predictions. So the real target of Pegasus is to perform and to guide killing experiments for the therapeutic project in order to validate our biological hypothesis on disease mechanism, on cellular models, on the real activity on the target in a given cellular model, in a given tissue, and so on, and also to gain biological insights for the biology. So if I ask Pegasus, okay, Pegasus, who are you? He will answer us that it's really a decision support tool for the early drug discovery process at Servier. It allows to capture and integrate meaningful and valuable data, internal and external, according to the business needs. One really important point, it's a discussion platform. And I think Martin told it during his opening talk. The graph technology, it's really interesting to put around the table different experts from different areas of expertise, and challenging a data model, challenging the question we can ask to the data model, challenging the data sources we have in order to answer the question, and so on. And what is really interesting when you are at the interface of different area of expertise, you have one simple question, but several answers. And as I said, so Pegasus helped us to design killer experiments and decide, and help us to design on experimental modalities, practical target. So is it better to target directly the gene? Is it better to target the transcript? Is it better to target the protein? And for the different application I showed you just before, we have planned experimental validations. So one, to inhibit protein aggregation for a neurodegenerative disease, where we use Pegasus to design a focused chemical library. Second, we are characterizing ASO off-targets using Pegasus for a rare and child disease. So the experimental validation are planned for start of December. And we are also de-risking the experimental modality, coupling UORF and ASO for a particular target. So we identify the different UORF, we design different ASO, and after we are planning to do the experimental validation in January of next year. So thank you for your attention, for your virtual attention. And I would like also to thank the different internal collaborators. So Pegasus, it's a collaborative project, basically. So here, main people that are participating to make Pegasus a good and a valuable knowledge graph. I would like also to thank Martin Preusse for the invitation, and also all the Neo4j team, because you are producing a really good technology, and voila. And if you want to contact us, Thierry or me, you can send us a mail if you want. Thank you.
Thank you, Jeremy. That was absolutely fantastic and great to meet Pegasus. It's incredible how many different data sources you have integrated, and how many different applications you have on that. So maybe one quick question that I find very interesting. So there was a question in the chat. Medicinal chemists often search for substructures. Is there a way to search for substructures or like chemical structures in general in your database?
Yeah. We are working mainly with our department of chemoinformatics and chemomodeling. And basically, they have plenty of algorithms that can subtract a scaffold of a molecule in order to rationally select some part of a molecule in order to have a particular activity, or also to inhibit or to decrease some features about the future molecule. So this is ongoing work about, is it better to generate this kind of substructure for the chemical structure in the data model before, so for the Pegasus generations, or is it feasible to calculate it on the fly with the Pegasus graph?
Mm-hmm. So it also has data model applications. So I think that's an interesting question. Thank you so much, Jeremy, for your talk. It was, again, absolutely fantastic to see kind of the depth of the data that you've integrated. And with that, I think we can move over to the next talk. As a quick reminder to the audience, we swapped the Health Echo talk with this one. And now Marjorie Kang from Northwestern University, from the Feinberg School of Medicine at Northwestern University, will present her project on mapping the OMOP common data model to Neo4j for pneumonia therapy response. So in general, we're kind of moving back from basic research and drug discovery into the healthcare space. So Marjorie, thanks for being here, and the stage is yours.
Marjorie, sorry to interrupt, but you are muted.
Apologize for that. Okay, I'm going to share the screen again and
Perfect.
okay,
Thank you.
Here we go. Okay, so here we go. Good morning or good afternoon or good evening, everyone. I'm Marjorie Kang, and I'm a clinical research associate from Feinberg School of Medicine, Northwestern University. And today I'm going to share a research in progress for a multi-year pneumonia project called SCRIPT. So here's our agenda today. I will first briefly introduce what SCRIPT school and its data sets we are collecting, and then I'll briefly introduce the OMOP data model that we use for the original DB and how we mapped OMOP to the Neo4j graph database for SCRIPT and our future steps. Okay, let's get started. So SCRIPT represents for successful clinical response in pneumonia therapy. And as we can see from this diagram, our center collects the blood and the bronchoalveolar lavage sample on a regular basis for critically ill patients in the ICU. And we have project one team focusing on understanding the successful host response to the infection, and we have project two team checking on the pathogen and its its genomic profile. And then we have data management and bioinformatics core, which I'm from, working on the data ETL and metadata management. And of course, we have the modeling core working on building predictive model to help us to tell ahead that whether a patient is likely recover or maintain the status or unfortunately expire. And of course, later we will verify the model with a second cohort and with the humanized mouse model. So, and a side note that not surprisingly that even though our project started before COVID, when COVID starts, we did see a big surge in the patient enrollment. And besides this main line of pneumonia research, we have, because of the pandemic, we have new branches like cytokines and COVID wave 1, wave 2, and the long COVID effects. But today we are mainly focused on the pneumonia successful host response and its OMOP common data model. Okay, so now we know what SCRIPT is about and many directions we are on. And here's a diagram that shows what data sets we are collecting. So besides this regular EMR data sets, we also collect REDCap survey data sets that use patient demographics information like patient history of disease or family history of disease. And then we have clinical trial management data sets and the Bell and pathogen metadata. And another big part of the data sets is the multi-omics that includes genomics, transcriptomics, and whole bacterial whole genome data sets, etc. So here's another illustration of how our data sets is connected. As we can see that from the right side, the OMOP EMR connecting through the clinical patients ID table that we put together, and then other enterprise data warehouse reports that we generated on the research as the research goes. And then through the accession number ID, which is the unique identifier for the Bell sample I mentioned previously, we were able to connect to the REDCap survey or clinical adjudication data to the sample level. And furthermore, with this Bell sample ID, we were able to connect to the multi-omics data sets. And of course, it's metadata like library preparation information. And today we're going to mainly focus on the OMOP common data model and how we mapped it to the graph database. So just a quick summary that how Script connects its goal to the graph database. So obviously, pneumonia is a complex disease, and we're trying to understand the mechanism of a successful therapy or the cause of the failure of patient recovery. And another big part is many clinical adjudications or decisions need to be made patient by patient, and that takes a lot of time of the physician's time. And we were hoping that with this knowledge graph that Neo4j database provides, we can help with the pneumonia adjudication process. And here's one of the many clinical questions that we frequently ask, that is whether this knowledge graph can tell us to distinguish a clinical CAP or VAP or HAP or non-pneumonia control patients, that is community-acquired pneumonia or hospital-acquired or ventilator-acquired pneumonia. And of course, from the technical part, even though we have the relational database, is called relational, but it didn't really tell us what the relationship between the EMR or the sample data sets or the multi-omics. So the graph database is a great potential for us to use the system biology approach to understand the host response and how the clinical phenomic data sets connects to the genomics level. Yep. And we are hoping to apply the graph algorithms to find any potential patterns behind the patient recovery or the disease development. Okay, so now I'm going to briefly talk about the OMOP Accommodated Model version 5 that we used for the relational DB we first started. So we, instead of including every detail in the in our relational DB, we started with these 9 standardized clinical tables, and including this healthcare provider table, and together with the vocabulary tables. And so far, we found that this already fulfills our need. And of course, thanks to the flexible schema of Neo4j, we can always add more data sets as we go, instead of have to drop the database as the relational DB. Okay, so now I'm going to share how we mapped the OMOP common data model to the graph database for our data sets. So as we can see from this graph that we kind of ended up with 16 type of nodes or entities or 22 types of edges along with its 55 node attributes. So the rule we followed is we wanted to put the nominal and categorical data into nodes, and then the foreign case in the tables, in the relational tables to edges, and then all the numerical values to either node or edge attributes. And as we can see that this our result schema is the entities, the subjects, the patient, and healthcare provider are on the top, and they are all connected to this instances table, like the observation period, the diagnosis, the labs data, procedures, observation, and medication data sets. And they are all connected to the encounters table too. And we also put the vocabulary table as separate nodes, and as well as the demographics datasets aside, because those will be the frequently queried datasets. So it will be faster than putting the demographic data in the attributes. I think what's worth mentioning is thanks to Jose, data scientist from Neo4j, we were suggested to have this next relationship, sort of self-directed encounter data sets. So we are able to build a patient journey on top of this. Okay, so that's the schema we got started. And here's a quick screenshot of what the data sets, the schema looks like in the Neo4j browser. As we can see that indeed the person and encounters table are the popular nodes that almost every other type of nodes are connected to the person or encounters table. And talking about how many nodes and edges we ended up with, that's about 7 million nodes and 15 million edges for our, as of October, enrolled patients, that's 553. Okay. So now we know that it's loaded into the database. So how's the data sets, like the statistics look like from the detailed node side by labels type? So as we can, not hardly to imagine that the labs data takes the dominant of the node counts, and then followed by the concepts, and then the diagnosis information and procedures, and then the encounters data. Okay, so now we got the sanity check that is aligned with our original OMOP DB, and now we're ready to try out some local or global analysis on this data set. So here's an example of one of the clinical questions that can we find the patients that has the spontaneous pneumothorax diagnosed and then has the maximum prescribed and also has the tube related procedure. So as we can see that we got 3 patients returned as the green nose, and each of them got admitted through either inpatient visit or outpatient or ER visit. But all of them has the maximum prescribed and the spontaneous pneumothorax diagnosed, as well as the tube-related procedures. And the query returns just in 3 seconds, and that's faster than what I imagined when we join 5 or 6 tables in a relational DB. That would require a longer query and longer time to get the results back. Okay, so now we know that the network works good for the local pattern matching. How about any supervised or unsupervised algorithms? Does it really tell us any hidden patterns behind the data sets? So we first started with the diagnosis occurrences. And we tried the node similarity and other community detection algorithms like Louvain or weakly connected components or LPA. But actually, not surprisingly, we found that the node similarity score between any 2 pairs of patients are very high. And as for the community detection algorithm, almost all of the cohort patient results in one huge cluster. And that's mainly because our cohort is ICU patients, and they are treated with similar antibiotics and get similar procedures, and of course, very similar diagnosis. So this, without any filtering, these algorithms didn't give us meaningful results. So we went onto a pipeline to continuously adjust our filter and apply some edge weight on the patient-to-patient shared diagnosis. And here's what we did. So we first did the filtering on the diagnosis we want to consider. For example, originally we included the past 5 years of diagnosis occurrences, and that's way too much. Then later, we ended up with only using the time point from the patient admission to the 2 days past patient intubation. So that really shrinks the diagnosis period to consider. And then we calculated the term frequency inverse document frequency score based on the shared occurrences and put it as the edge weight for the patient-to-patient shared diagnosis. And later on, we went through the regular steps like build subgraphs and adjust the parameters in the algorithms, graph algorithms. That's the GDS, Graph Data Science Library, we were using from the Neo4j plugin. And then we were able to apply the Louvain algorithm and do some further exploration of the communities. Yeah, and here I forgot to mention is we also did distinguish between acute and chronic indicators because many of these ICU patients got intubated or got admitted to the ICU or intubated because of acute diseases. So we want to filter those too frequently shared acute diseases to rule out those like network. Otherwise, we will have a very dense patient network, and that doesn't give us much value in terms of clustering results. Okay, so that's pretty much our pipeline works. And here's the results we got so far. So we did find 5 distinct communities, and excitingly, each of the community has a dominant type of disease group like cancer or respiratory disease or chronic diseases. But when we further want to match these communities with our pneumonia episode types, that's what I mentioned, the CAP, VAP, HAP, or non-pneumonia control group, it didn't really match the group counts as we can see from the right table. So obviously, it doesn't necessarily mean that clustering algorithm doesn't work. It's just it didn't give us the classification results as we expected, and we still need to do some exploration on how these 5 clusters are telling us. Maybe that's the potential patterns that the network, the edges give us. Okay, and here's our next steps. So originally we used the term frequency inverse document frequency score as the data cleaning steps, but now we got the suggestion from Jose that the degree situationality algorithm is another good tool to roll out those highly popular nodes so that we can have a less dense network to run our graph algorithm. And another one is we want to try the node similarity score again, but this time on a bipartite graph instead of a monopartite graph. That is the patient-to-patient shared network we tried. And of course, we want to add more features like the REDCap, demographics, and clinical adjudication datasets, as well as multi-omics datasets. And another very interesting path would be the patient journey, that we can model how the one patient evolved from one visit to another, and how the labs and procedures and diagnosis are linked together. Okay, so I think that's pretty much what I would like to share, and I would want to express my great thanks to my awesome team and Jose, data scientist from Neo4j, and other Neo4j enthusiasts like Dave and Narciso. And of course, we would like to thank our founder, Nayad, and Neo4j for providing such wonderful platform. And with that, I would like to thank everybody for your attention, and I'm happy to take any questions.
Fantastic. Thank you so much, Marjorie. That was, I think, a really, really interesting presentation, especially because I really like this one slide where you had OMOP on the top right, but also multi-omics and so many different data sets. I think you really showed what kind of scope a graph database can handle. Let's have a look at the questions. Let's start with something technical, maybe. So Derek Wong is asking, do you use a single computer or a cluster to host your Neo4j database? And how much RAM do you need to run those analyses?
Yeah, that's a great question. So originally, I started the test on the local desktop, but then the performance is very poor. I remember I first started on a 32 gigabytes RAM, and one time it took me almost 8 hours to load the data sets. But then later, Feinberger School of Medicine IT was able to set up a server, on-prem server with an encrypted connection. I believe the RAM is pretty large now, 256 RAM. But I'm pretty sure the enterprise version might benefit, like maybe compressing the database a bit more, because currently we are still using the community edition. And in terms of database size, it was actually kind of large. Currently, for those 550 patients, we ended up with 80 gigabytes for the nodes and the relationship store. So we haven't really dig into optimizing the site store, but I'm pretty sure there's ways to optimize the index and the relationship stores.
Okay, so you do have a pretty big machine, but you can run very complex analyses on that single machine. Okay. There's another question. How do you claim or decide that the clusters that you identify are really significant?
Yeah, that's a great question. So in terms of the clusters, I think after we found that the clusters didn't exactly match the pneumonia episode types we desired, we moved on to like checking the ICD-10, I believe, ICD-10 groups. I remember there are 21 like larger disease group category. So we sort of draw some Manhattan graph to see whether the communities has a dominant match with any disease group. So that's what we went so far, and we sort of park a little bit after that exploration, and now we are shifting back with more like classification algorithms, with the clinical adjudication labels. So sort of switching from unsupervised to semi-supervised.
Okay, I hope that answers the question. And as usual, if you have follow-up questions, want to clarify something, just post a message to the chat. I would also have a question, maybe.
Sure.
So the OMOP common data model is a common data model, obviously. And do you think it's possible to generalize your graph mapping of the OMOP CDM? So do you think it would make sense to come up with a unified common graph data version of OMOP?
Yeah, that's a great question. That whether we can apply our center's results to share with outside, like who doesn't necessarily have the similar graph. So I can see that as long as the center has the same OMOP accommodated model structure, they will. I actually uploaded the code to build up the database, so I'm pretty sure they can just even retake the code to just build up the database in few minutes.
Mhm. Okay. And maybe as a follow-up question, have you reached out to Odyssey already and talked to people in the Odyssey community about a graph representation of OMOP? Are there other people working on that?
Yeah, so I think one of our my colleague who is highly involved with the forum, but I personally haven't reached out. I remember I posted one question when we were working, when we at the beginning to see whether someone else working on it, but didn't got much responses as we want. So we kind of started our own journey on that. And we are planning to share this in the paper. So probably more people will be able to reuse this method and doesn't have to create their own wheels.
Mm-hmm. Yeah, I think that would be very interesting, because I think in a similar way, how we need this, basic gene and protein information in every pharma project, we need these vocabularies and terminologies and all this in every medical informatics project, right? I think some collaboration between Odyssey and knowledge graph communities would be fantastic.
Yep.
Let's see. So, Marjorie, thank you so much for your presentation. I think that was really, really interesting. I can't wait for the paper. I can't wait to see how you integrate multi-omics and the survey data and everything. So,
Great.
Thank you so much for your presentation.
Thank you for the invite. Thanks, everybody.
And I think with that, we can hand over to Claire Simpson for our next talk. Claire will describe her method and publication. So this was published, I think, in May this year, about analyzing perturbed co-expression networks in cancer using graph database. So Claire, the stage is yours. Looking forward to your presentation.
Thank you. So yes, I'm going to talk today about analyzing perturbed co-expression networks in cancer. The original motivation for this project was basically twofold. First, because gene interaction networks look and basically are just graphs, I was curious to see if using common graph algorithms on them would yield any sort of biological insight. And secondly, to focus on the more specific question of, what happens in cancer? Will these graph algorithms show us that these networks have been perturbed or changed, and what sorts of changes would we see? So I'm going to talk briefly, first of all, about the input data that I used to create the network, how I built the graph using Neo4j, 3 different classes of graph algorithms used to analyze this data, and then just a brief discussion about what I found, what work could be done in the future, any questions. So I started off with data from the Cancer Genome Atlas, which is a very comprehensive, super useful resource. They have all sorts of data, but I was looking specifically at RNA-seq data from these 6 different cancer types, which were chosen because they had a significant number of patients who also had contributed data from matched normal tissues, as well as tumors. So I took data, this RNA-seq data from these 6 cancer types for only the patients which had tumor and matched normal data. Then I calculated the correlation coefficient, Spearman correlation coefficient, for each pair of genes in both the normal and the tumor networks for each of these 6 cancer types. I used the Spearman coefficient because it was more outlier resistant than usual Pearson, and basically chose a cutoff of about 0.8, and in order to create sort of a scale-free power law distribution for the resulting networks. Here you see sort of a pair of genes and what these correlations sort of ended up looking like. If we take KRAS and ZNF 800 in the normal tissue for lung adenocarcinoma, you see that there's like a pretty straightforward correlation between them. Each point here is one patient and their levels of KRAS and ZNF 800. So yeah, in the normal, there's a good correlation. And then you are already seeing in the tumor that something has sort of gone awry. There's, first of all, lower levels of both of these genes in most patients, but then also when you do see higher levels, the connection between these two genes has sort of disappeared. So in this normal graph that I would make, there would be an edge between these two nodes, KRAS and ZNF 800, but there wouldn't be such an edge in the tumor graph. So then I sort of imported all of this data into Neo4j. I created one node per gene. And then if the correlation coefficient in the matched tumor tissue was higher than my cutoff, I added a match edge, an edge labeled match. And if it was above the cutoff in the normal set, I added an edge that was sort of labeled normal. And I made a Neo4j graph for each of the 6 cancer types. I also annotated each of these nodes with gene names as well as accessions, and later would annotate it with results from the graph algorithms such as PageRank or communities. So this is essentially what it would look like in Neo4j if you were to visualize it in Neo4j. Pretty much immediately, these the matched tumor and the matched normal networks started to look extremely different. The number of nodes which had any edges at all in the normal versus the tumor like subgraphs were much higher in the normal versus the tumor. So there was already sort of a loss of interaction that was immediately visible in the tumor tissues. This was even more pronounced if you looked at the number of edges, especially in breast cancer, which is BRCA here. Number of edges in the matched normal subgraph was extremely high, but in the tumor subgraph, which is taken from exact same patients, the exact same method of like preparing the graph is like very strikingly lower, showing that there are interaction networks that have been disturbed. So then we wanted to look at more specific genes and interactions, and used several different algorithms that Neo4j has implemented in order to do that. So the first thing that we looked at was calculating a PageRank score for each subgraph and each cancer type. Basically, this is the same algorithm that Google invented and uses to some degree still, in which a node is sort of, its score is voted on by its adjacent nodes. And for example, a node that has a lot of connections has a more powerful vote. So the central node here is going to vote on the leftmost node to raise its score, and its effect on the leftmost node score is going to be higher than, say, this top node's effect on the leftmost node score. So once we calculated the PageRank for each gene in both of the subgraphs, we were able to find a few different things. First, we observed that the genes that had the highest PageRank scores were often known master regulators. In the lung adenocarcinoma normal subgraph, most of the top 10 PageRank scored genes were zinc finger transcription factors. And these, most of them, lost their incredibly high PageRank scores in the tumor subgraph, which was very interesting. Then what I wanted to do was compare between different kinds of tissue types. So if we looked at the correlation between a PageRank score for each gene between, say, like 2 different kinds of normal tissue or 2 different kinds of tumor tissue, the correlation was much higher between different types of tumor tissue than it was between different types of normal tissue, which suggests that the important central nodes with high PageRank scores tend to be not very similar between normal tissue types. For example, like an adjacent lung tissue and an adjacent kidney tissue. But these important central nodes between, say, like lung cancer tissue and kidney tumor tissue, those might start to share these important regulators. If you'll note, there is an outlier in the normal half of this graph, and that's actually between the 2 different types of lung, so lung adenocarcinoma and lung squamous cell. So those are actually more similar, which you would expect. Next, I want to move on to talk about Louvain clustering and community detection, which basically Neo4j uses to calculate its communities, for example, like this in this very small subset of the whole graph. The immediate result from this analysis was that the average community size decreases significantly in tumor types, which you might expect because there are also just fewer nodes, fewer interactions. That should naturally lead to smaller communities, which is true. So then, looking more closely at the specific communities, we ran a gene ontology analysis for each of the resulting communities, in particularly the lung adenocarcinoma set. The interesting thing here was that if you look at the change in degree, which is another measure of centrality, you see like vastly different behavior between different communities. So it's not like genes are uniformly losing or gaining connections. It's happening like specific to different subsets of the graph. And additionally, we found that these subsets, these communities, they are biologically relevant. So for example, this community 4 here is highly enriched for genes involved in DNA replication. And what we found there is that these genes involved in DNA replication as a community, they tend to actually gain different connections in the tumor set, which is very strange because in general, connections are lost in tumor. An additional group that gained degrees, gained connections in the tumor set was this community 15, which is enriched for immune response. And so looking at these specific communities, the genes in these communities that have gained degrees can be used to identify pathways and genes that are involved in cancer, and in fact, like increased enriched in cancer. The last graph algorithm that I'd like to talk about is this shortest paths algorithm, which is pretty straightforward. You just calculate the length of the shortest path between every pair of genes in the 2 different subgraphs of each graph. So for here, it would be between this bottom left node and this bottom right node. Shortest path would be 2, not 3, not 4, but 2. And wanted to see if the distribution of the shortest paths was similar or different in tumor versus normal tissues. And so as you would expect from the loss of edges in the tumor set, the path lengths tended to increase in tumors because these connections have been lost. The paths between different interacting genes need to get longer in order to be connected. And so this is another sort of bit of evidence that first, we are sort of seeing something biologically happening in these graphs, and secondly, that there is a perturbation happening. So there's a lot to go from this. We were able to identify lots of like genes, subnetworks, types of genes, communities, which seem like they could be involved in this sort of process of perturbation. But something that is very interesting and was not sort of able to be studied using this TCGA input data was that adjacent normal tissue is not necessarily itself normal. And it would be extremely interesting to construct a co-expression network and graph from like truly normal tissue, because adjacent normal tissue might have some tumor cells in it. It might be having an immune response. So what would we see if we compared that to normal tissue? What would we see if we compared like really normal tissue to tumor? Another thing is we were looking at RNA-seq data, and I think it would be really interesting to compare that with the same co-expression network as generated using proteins and mass spectrometry data, for example. Would the same networks and important nodes emerge? Would it be possible to do similar things with signaling networks, for example? Just a really interesting question for the future. And finally, I was wondering, how does this co-expression matrix correlate with like known interactions? Certainly what we do know now is relatively sparse, but do the genes that we know to interact and to be correlated, do they show up in this graph already? And so we do know that like a lot of the genes which are known to be involved in cancer did show up in terms of like central nodes, for example, or significantly decreased or increased interactions. But yeah, how does it relate? And I'd finally like to say that using Neo4j to create this graph, analyze it, and visualize it was extremely fast for all steps of the process, like almost instantaneously calculating PageRank or communities. And it was very technically straightforward. So I'm very grateful to Neo4j for providing this software. And I'd like to thank my company, Cell Signaling Technology, because this is where I did all this work, and especially to Florian Nad for advising the project, being very instrumental to it. So thank you so much. If anyone has any questions, I'd love to hear them.
Fantastic. Thank you so much, Claire, for the presentation. And what I find really interesting is that you showed so many examples, essentially, of very detailed analyses you can run on a graph. Let's see if we have questions here. So there's a question from Thomas asking, did you miss any algorithms in Neo4j that would make sense to analyze tumors?
That's very interesting. So I think that these are the main three classes of algorithms, sort of centrality, shortest path, community detection, but I definitely didn't use every type of each subset of these. So for centrality, I was really only using PageRank and degree. For community detection, only Louvain, for example. So there's definitely room to look at what other sorts of community detection algorithms exist and how they would work. I would say especially community detection would be interesting. Yeah.
Mm-hmm. Okay, that's a hint maybe for the Neo4j product team. What should come next? Maybe a quick follow-up question. So you focused on algorithms. Did you also look into the predictive parts of the GDS library?
Oh, not at all. That's very interesting. I couldn't hear the second half of that, but I heard predictive.
Sorry, yeah. So the GDS library also has predictive capabilities, right? So there's node classification methods and link prediction, and you can calculate embeddings, which you can then potentially use to build models and all that. Do you have any plans in that direction?
So I've been working on sort of a different set of projects ever since completing this, but that is super, super interesting. And I want to write that down and see just what that resource is.
Yeah. So yeah, the GDS library, I think, is expanded, has expanded a lot in the last months or in the last year or so, and lots of additional algorithms have joined. So let's have a look at the chat and at the Q&A. I think there are a couple of questions here. So there's a question from Derek. Does the difference of number of samples between normal and cancer affect the calculated results?
It definitely would. So I only used the just the patient data where they had both. So for my analysis, they were always the same number because if there was a patient which only had tumor data, I excluded them from the analysis. But I certainly think it would affect it, yes.
So there's an additional comment. I have the impression that the number of normal samples is much smaller than the number of tumor samples in TCGA.
Yes, that's true. Like with the previous question, that was why I could only use these 6 cancer types because they had, I think, above 50 or 60 normal samples from those patients, and then I excluded patients where there was only tumor data.
Mm-hmm. And I'm looking at your graph and at the data analysis. I would like to recycle a question that we had earlier. How do you update the graph, or did you already update the graph with new releases of some of your data sources, and how would you approach that?
I have not done that yet. If I were to do that, and that is an interesting question, I would probably, because this whole process from the beginning was quite straightforward, just calculate the co-expression matrix, check to make sure my cutoff was still appropriate, and then re-upload to Neo4j. Yeah.
Mm-hmm. Okay. Because I think in many cases, like when working with genes and gene IDs and so on, all this changes. And I'm just asking because we, I mean, in many of the talks, we have similar basic data sets. We all need genes and proteins and so on. And I kind of want to force everyone to collaborate on this and share resources and share ideas how to model this and how to load this into a graph. But we have more questions from the audience. Piers is asking, how many genes in your observations are affected, and affected is reduced or increased? Connections between control and disease. I think I can read it again. Does the question make sense? How many genes are affected and are connections between control and disease?
I don't remember exact numbers, but I think, yeah, the majority of interactions were lost in tumor networks versus normal networks for all different cancer types. So it would be most of them. And I actually didn't sort of, even though they were in the same graph, I didn't sort of analyze both tumor and match at the same time using Neo4j. And I would wonder how that would work, what sort of analysis could be done with that.
And there's, I think, an associated part of the question. How did you reduce the number of genes to study?
I reduced it, I guess, using only the cutoff for the correlation, for a significant correlation. So if a gene didn't have any significant edges in either the tumor or the normal tissues, then it wouldn't be included in the Neo4j graph. But otherwise, I took everything.
Okay, so I hope that answered the question. If not, just post another message on the chat. Let's have another look at the Q&A. If there are more questions coming up, I think that was pretty much it. I would have one additional question, also one that I asked for a couple of other talks. So obviously you use GDS a lot for your data analysis, right? What do you think, next to the question which other algorithms should be in GDS, how did the workflow work for you to get bulk data out of Neo4j into other, data analysis frameworks or tools or applications?
So I found the sort of going from the back of that question to the front that if I wanted to, for example, export like all the PageRank scores for every gene, that was extremely easy to do by using within the call to PageRank itself, asking it to annotate each node, and then exporting just a straight table with the name of the gene and the PageRank score in each of the subgraphs. I believe that question also contained something about the workflow for setting it up, and that I remember mostly being very simple, but having to work around the large number of genes that I was inputting. So I needed to call it in sort of a loop to get it to work.
Mm-hmm.
And I think I might be missing some of the middle of that question too. So if you could repeat that.
I think that was a good answer. So it's something I genuinely wonder about how people approach this. GDS is great, but GDS cannot solve every problem. Getting data out and potential results back in is just, I think, a workflow that a lot of people use, and I just like asking the question. But apparently it worked for you, and so the description of the workflow was a very good answer to that question.
Yeah, I think that the total size of the graphs was much smaller for me than it would have been for the previous presenter, because I was just looking at the size of the exported graph itself, and it was all less than 100 megabytes for each of the cancer types. So a little bit easier for me than maybe for some.
Mm-hmm. Mm-hmm. Okay, so let me check the chat. I think that's it. We don't have any more questions. Since this is published work, you can of course all look at the publication and look for more details and potentially contact Claire for more questions. So all I can say is thank you very much for your talk, for your presentation. It was fantastic having you here.
Yes, thank you for having me.
And Alex, our next speaker, is around already.
Yes. Meanwhile, you can see it's gotten dark here in Munich, but yeah, Evelyn is in backstage. She just needs to switch on her webcam, I guess, and then I can I can let her in, basically.
Fantastic.
Yeah.
Because I'm really looking forward to our last presentation, to our last talk from Evelyn Travnik. About the Lifeline platform, a graph-powered knowledge reconstruction and mining platform to accelerate research. And I've said that a couple of times during the day. I've been looking forward to this talk because I think this is a very comprehensive and massive platform with a lot of analytics going in. So I'm really looking forward to your talk.
Hello, everyone. Hi. Can you hear me okay?
Yes, we can hear you.
Okay, great. Let me try to share my screen. Okay. Oh, no. Sorry, we have a bit of an issue here with sharing. Apparently, I do not have sharing permission set up. Give me 1 second, please. Yikes. I'm new to using a Mac, and so there's a lot of permission settings here.
It always happens when you have to set up a new computer, allowing access for every tool, every platform, every website.
Does anybody know, by any chance, system settings to allow sharing? I don't know why that's disabled. In Zoom, it's never disabled. So sharing.
Yeah, good question. I think you need to enable Restream to be able to access your screen, but I think there should be a prompt, right? If you do it for the first time, it should tell you, do you want to allow this, and then you need to click yes or something.
It says unable to share the screen. Please update system permissions to allow screen recording for the browser.
Alternative, you can also send me your slides by email, and I can show them.
So, yeah, maybe we could just take a minute break. I mean, still 5 minutes early, so sorry. And we can look at it.
All right, let's do that.
Perfect.
Should we talk about something while Evelyn sorts out her screen sharing, or worst case shares it with us, and we can then use it here? Or does everybody want to use? I mean, we are basically, this is the last session right for today, so maybe everybody could use a short bio break if you sat through the 3.5 hours content till here. Maybe everybody is a little bit. It's good to stretch your legs and move away from desk. But
reduces your risk for thrombosis.
Yeah,
to get up every now and then and walk and move around. So that's important.
It is.
We can still try to talk about things. Can someone in the chat suggest a random topic?
Yeah, exactly. Yeah, that's a good thing. We don't want to. I don't want to. Advance the recap, which we will do obviously after Evelyn's session. But if you have in chat anything you found most interesting today, or what was your highlight of today, type it in. We'd be interested to hear how you thought of the day. What was your highlight? For me, because I maybe I won't be. Michael will join the recap, but I can share my highlight, which was definitely Jeremy's presentation, the Pegasus project. Even though I don't have a biological background, so I understand only maybe, I don't know, the tip of the iceberg. So let's say I give myself I understand 20%, but I could really tell that this was really a cool project and really interesting to see. If you look for this pattern, look for the connection between the various agents. That was, and I can imagine this is super insightful for any various use cases. Yeah.
Mm-hmm. Totally, can completely agree with that. What I find super interesting about that is that you can see how much power there is on the query level. I mean, we all want to do AI and GDS and whatever, but there is so much power in once the data is properly integrated. There's so much power on the query level. I think this is really interesting.
Yeah. And what do we have in chat? Abik says anything on graphs and imaging, like image processing, I guess, maybe. I think I saw something like this a while ago, where they use the images from various images from on a I don't know actually if it was medical related or something else, but they used these and then stored metadata in Neo4j to then relate images that had the same metadata information with other images, and then they could, I think it was medical related, and then they could find out if an image was tagged as cancer, and then another image that looked similar didn't have that tag, they would flag it and then would find out, oh, maybe actually this image should maybe have the cancer tag as well.
But I think the question is interesting because imaging in general, or image analysis, is one of the fields where I haven't seen real world publications and Neo4j applications yet. One is imaging, and another thing that I think is super interesting, would potentially be super interesting, and we kind of had a question about the substructures and all that. So far, I haven't seen many publications on using graph databases for actually storing molecules. Where a single atom is a node, and how to model chemical conformations and all that. So I don't really know anything in that direction. And I don't know paper where they use graph databases to store. Is the thing that you mentioned, is that published somewhere?
Sorry?
The project that you mentioned, is that published somewhere?
It must be. I must have seen it somewhere, but I don't remember where from top of my head. I might be able to find it, but yeah. All right. But meanwhile, Evelyn pinged me and said she's ready. If yeah, I think we are ready. I'll can slide her in, and then we can listen to Evelyn's presentation.
Fantastic. Looking forward, Evelyn. Thank you so much for being here, and the stage is yours.
Yeah. Hi, everyone. Thank you for having me. Let me also put my presentation in presentation mode. Okay, can you see my screen now?
Yes, we can. Thank you, Evelyn.
Okay. Yeah, perfect. Great. Okay. Yeah, so I'd like to thank Neo4j for inviting me today to present. It's a pleasure to be here. I will be introducing you today to an open source knowledge mining platform that I'm developing with my team. And this platform is based on Neo4j. I am currently the Chief Information Officer at the Center for Biosustainability. We are a research center that is funded by the Novo Nordisk Foundation. And we have about 300 researchers that represent over a lot of different nationalities. I think like over 47. What we do at the center is we engineer cell factories that we use for the production of things like sustainable chemicals, natural products, such as therapeutics, also microbial foods. We have a program on that. This is where we look at like new food sources like alternative meats, therapeutic foods, etc. And we have also funding programs with focus on infectious diseases as well as microbiome. I lead the informatics unit with a team at UCSD in San Diego. This is where I'm based out of mainly. And I have a team at the Center for Biosustainability in Copenhagen, Denmark. This is our headquarters. The mission in our team is we want to develop systems that can turn big data into knowledge and do that with utilizing artificial intelligence, natural language processing, and knowledge graphs. Ultimately, we want to enable people to make sense of their data and establish deep insights from their data. So a little bit about myself and the portfolio of products that I've developed throughout my career. I've built a lot of like different enterprise level platforms over the past 15 to 20 years. This includes things like computational modeling, as well as like sort of like pathway prediction for the production of biochemicals and the ability to like generate IP. And a lot of these things that I've developed, they could have really benefited very much from graph approaches and graph databases. But back then, graph databases weren't very popular and weren't able to do that. But so I have a lot of like background having sort of struggled with old technologies. Now, fast forward to today, we're building Lifelike, and here we are able to take advantage of the power of things like Neo4j graph databases. And so let's take a look what Lifelike is. So as I've already said, it's a knowledge mining platform. But beyond knowledge mining, we can also use it for knowledge reconstruction and very much for data storytelling. And so I will show you very much how this can be done. We utilize artificial intelligence, knowledge graphs, and Neo4j and graph algorithms. What we can do is we want to be able to understand unstructured or structured data quickly, bring it into Lifelike and quickly understand it. We want to be able to extract key information in a visual format, and we can cross-connect that knowledge with other sources and do that. And then with graph algorithms, we can provide deep insights. We also like to talk about, besides knowledge mining, we also like to say we're supporting graph data science. It's also a term that's kind of become popular, right? That graph databases are enabling. We're able to go from data science to graph data science. So Lifelike is powered by Neo4j. We populate it with many different public knowledge sources, things like Biopsych, UniProt, etc. You know, a lot of these things I'm sure you're going to recognize. And we also populate it with important experimental information from our internal databases and data lakes, etc. So essentially, we are building kind of like a digital mind type of an infrastructure here that is powered by Neo4j. and knowledge that is stored in a better format for us to be able to better process and to be able to gain a deeper insights from it. I believe that any organization needs this type of an infrastructure, that we cannot afford not to invest in this because it is very powerful. And also, I think Gartner has said that by 2025, graph technologies will be used in about 80% of data and analytics innovations. So they're also predicting that there will be a steep growth in this. And I agree with that. I think that every company needs this. And when this is done well, this is really providing people with a competitive advantage of being able to build kind of infrastructure that resembles a digital mind. And this type of an infrastructure allows us to really develop kind of like a virtual assistant type of an application, and therefore also the name Lifelike. This is really what we'd like to get to, the real like a assistant for scientists. So where does knowledge mining and graph data science fit in with other analysis techniques? So here we have a graph where we see different analysis techniques that we have available today, ranging from black box to white box. On the white box side, we have full mechanistic modeling, and this is something that I've been doing a lot in the past and built platforms around that. So this is very powerful. However, you cannot do it for an entire cell. You can maybe do it for metabolism, but you cannot do it for the regulatory system and many other things, right? So it's limited in scope, yet very powerful. Then what happened when people used to do that also, for example, in fraud detection, right? Mechanistic modeling, very popular. But then when data exploded and became cheaper to generate, people jumped to machine learning. And machine learning is very much black box, right? We get back a result, but we don't really understand why did we get back what we got back. And so we can't explain it. And what lies in the middle here is knowledge graphs and graph databases. What we can do here is really take our black box results to another level and talk about explanatory AI, right? We can start to understand why are things related. And we can now bridge the gap between the black box and white box and analysis techniques. And so as you can see that there's a strong focus on knowledge management, right? We can only do this if we manage knowledge properly. Only then we can tap into prior knowledge. So knowledge management is fundamental. And let's take a look. How do we manage knowledge today? We first start with generating data, which we put in well-established things like databases and data lakes. And then we analyze the data with many different analysis tools. There's an explosion of tools available for that. And then once we analyze the data, we generate knowledge. And where does the knowledge go? It mostly actually flows into text, right? We put together PowerPoint slides, PDFs, we publish, maybe we have an ELN where we talk about it, maybe we talk about it in a social media channel internally, the company. So a lot of it goes into text. And so now we're having a really hard time to reverse engineer that knowledge, right? And be able to use it again. And the way we can do that is with natural language processing. So this is one of the hardest AI problems, but we're here then left with reverse engineering it again from text. Sometimes we also put knowledge bases back into relational databases. In that case, I would say we put it back into data. We shouldn't talk about knowledge, right? So it's very complex schemas we put it back into. And then it's really hard to understand these complex data schemas. And here we're finding that natural language generation can actually help us. And so we've been starting to do that. So again, here, let me illustrate this further. Again, a majority of knowledge ends up in text. So then we use natural language processing to reverse engineer it. Ideally, we want to extract the relationships between entities that are discussed in text, as well as like getting the meaning out of it. So basically, we're trying to extract mechanistic relationships. When we mine structured data, that can be very complex. For example, Reactome being one such example that we have been working with a lot. So when we're mining that, it's very complex schema. So it's really hard to understand the relationships. So we've developed techniques that are helping us to collapse knowledge, to like visualize it in a better way, to reduce the complexity, as well as use natural language natural language generation actually to explain to a user what they are seeing. And this allows us to understand complex relationships. So a better way to manage knowledge is with the use of knowledge graphs stored inside a graph database. So this format is easy for a computer to process, as well as for human to follow and understand. Now let's take a look what we can do once we have established these capabilities, right? So if we have this large integrated knowledge base that consists of many data sources, what can we do with it? And some examples are, we can do semantic analysis on it. So we can process text and pull out the entities that we know, that the knowledge graph knows about, right? So we can pull those out of text and create relationships between them. We can also take a data source and enrich it against this knowledge graph. So we can, on a button click, pull out very quickly information from many different data sources, as well as like if our knowledge graph contains ontologies, we can also take a data source against that and do a statistical enrichment. And then finally, of course, we can take a community of entities, for example, let's say a community of genes, and we can use graph algorithms and analyze it against this knowledge graph. And finally, once we have such a knowledge graph, we do need to be able to like update it. So we also have the ability to do knowledge reconstruction, and as well as like extract knowledge from all these analysis techniques. So basically do data storytelling. And I will now show you examples for each of these. So the first one being natural language processing. So here you see a picture of a document that gets uploaded into Lifelike. And the nice thing is you can actually upload any PDF. So anything you can save as PDF, you can bring into Lifelike. It could be a website that you save as PDF, could be a PowerPoint slide deck, could be a drawing of something. So many different things you can bring in. And what we do then is analyze known entities using our knowledge graph, and we color code them, and we link them back to the original sources where we can find more information about them. We can also click a button and see what are all the things that are being discussed in this document. And when you click here, you can navigate through the document. Let's say you wanted to narrow in on something particular. We are also actually annotating content using our own internal sample IDs. So for example, here, this is a Jupyter notebook saved as a PDF, brought into Lifelike. And then here is a graph that contains sample IDs from our LIMS. And so we can now, we can recognize that this is a sample ID. And when you click on it, it takes you back to the LIMS, and you can see the metadata for it. So it's a great way to integrate databases and enrich the knowledge. We're using here AI, but we're also doing rule-based annotations, which means that if you have a favorite term, you can highlight it here, and you can now annotate it on the fly. And from now on, it will recognize that term. So then knowledge extraction and enrichment. That's another example what we can do with the graph. So let's take a look at some of these examples. These are enrichment tables as well as statistical enrichment. So here's an example where we have a list of genes. This was a list of genes that was brought back from a AI analysis techniques. Basically, we got back said this community of genes is supposed to be regulating maybe iron, and now we're trying to understand, but how does it do that? What is this community of genes doing? Sorry. Previous was too fast. So on a button click, we get now information from many different databases extracted. And what we do also on top of that, anywhere we find text, we always do a semantic analysis. So not only can you see the text, but you can also see the entities that are extracted from it, and then color coded again. And then on a button click, you can also do a statistical enrichment, in this case against the Go classes. And so what we're seeing here at the high level, we can quickly grasp what are these community of genes about. Its enterobactin biosynthesis kind of stands out here, right? And so the other thing that we have done, though, instead of just looking at the text we get from databases, we said, well, there is other abstracts being, publications being referenced. What if we take those abstracts also and pull them in and do a semantic analysis on it? And so at that point, we're seeing kind of similar results. We're seeing maybe a stronger signal because we have more text to work with. But if we're now, in addition to just extracting the entities, also extract relationships between the entities, then we are able to make a discovery here where we saw that there was only one metabolite that stood out that was related to iron starvation, growth, and oxidative stress. And because we have able to now extract these relationships, right, we're able to see that only one metabolite is connected to those three. And suddenly, when it was so small over here, right, suddenly it becomes to the foreground and it comes to your attention. And so this is kind of like finding a needle in a haystack, right? So this is an example how graph technology and extracting relationships can help us see new things that we didn't see before. Here's another example how we can connect the dots. This was a document where I took the entities out of the document. On a button click, you can do that. You can take the genes from those genes, you can create an enrichment table, so grab a lot of information from the knowledge graph. And when you looked at that entity cloud, something stood out, right? And that was not discussed in the paper. So something else stood out that we have now enriched, brought to our attention. And I was able to see that and make a connection, remember, and search within Lifelike and see that, oh, this also showed up in one of our other analysis techniques where we did a tracing against the network. And so now I can make a new connection. Knowledge mining. What have we built for knowledge mining and graph algorithms? Let's take a look at that. So here's the approach that we are taking. Let's say we want to analyze two communities, and one community might be a community of network nodes. So one community might be, let's say, up and down regulated genes that were measured under a certain experiment. In this case, cells were exposed to catecholamines, so stress. And so we measured the up and down regulated genes. On the other hand, we also had measurements for the plasma metabolites. And so now having this two community of entities, we want to go against the graph and understand what connects these two communities. And we can do this against different knowledge sources. So for example, we can do this against biopsych, we can do this against the literature data set, or we can go against Reactome. And so when we do that, the way we do this, we then applied a graph algorithm that was a combination of, we tried different things, PageRank, as well as like in-betweenness, customized in-betweenness analysis. And so then we ranked basically the entire graph, Reactome graph, right, that was highly connected to our source and destination. And so then we end up with like a ranking result of network nodes. And then we can ask the question, how are certain things, first of all, this result in itself is very interesting to look at. And then we can pick and choose what we want to see the relationship to. So let's say then we take one endpoint and say, show me the connectivity from that endpoint to my starting points, to my source and my destination. And so we're doing this in this way, we're really able to like detangle hairballs because we're able to see snippets at a time. It would be impossible to see this all in one graph, right? But because we can narrow in on snippets at a time, we're really able to detangle and understand the mechanism, understand what connects, and make new discoveries. And we've seen really great things with this approach. Unfortunately, I cannot talk about any results because they have not been published yet. So here's a little bit of Lifelike in action. I want to just show you how you can navigate through it. So here's one of these visualization techniques. Sankey-like visualization techniques. We feel like this works really well for detangling hairballs. And so you can navigate through this, you can click on things. And we always have the ability to show the shortest paths from a particular ranking point back to our starting points, or do the paths of highest influence. So basically we've implemented these techniques where you can switch back and forth to see different things, just the most relevant things or all of the shortest paths. So so a couple of examples here, what this might look like. Then an example on data storytelling and collaboration. So what I have here is, you can really use Lifelike to pull together, if you have a user meeting and you want to talk about some results, you can pull together a map, draw a map in Lifelike that pulls together the entire story. And so let's take a look how I've done this here for one of the user meetings where we discussed results from a particular analysis. We had run these traces. And so from this, I can use this map to navigate through my results and pull up these results from this analysis. And so basically you just click on this item in the map and you open up the corresponding other content. And it's a really nice way of navigating, and you can pull your entire sort of data story together here in Lifelike. You can also bring in other file types, for example, Excel spreadsheets. You're not going to be able to open them up in Lifelike, but you can download them. You can still house them in Lifelike, so you can pull together all the information. So here's another example how we are using the knowledge graph, the literature knowledge graph, actually, to enrich our knowledge here. So here we narrowed in on one particular gene that we're interested in, and then we want to ask, can I find more information about this gene in our knowledge graph? But what's the literature saying about this gene? So I can now double click here and I can open up all the associations for all the associations with chemicals, as well as like diseases that have been discussed in the literature for this gene. And we're nicely clustering this here on the screen so that we're detangling again hairballs. We can then take a look at what are all the associated diseases. We can see that 75 snippets are associated with one particular disease, etc. So a quick overview. We can also open up these snippets and understand which literature they come from. And when we are looking at this, we might also be interested now in grabbing some information from this knowledge graph that we're interested in that fits our data story. So you can actually just grab stuff again from here, drag it over onto your map and start storyboarding and saying, okay, I'm interested in this, right? And I want to capture this for my particular project. And you can just grab these things and also basically put evidence for any link that you have in the knowledge graph. You can grab papers and drag them over and provide evidence, saying these papers support this relationship between this entity and that entity. So you can also grab these things and just put them on the canvas if you'd like to see the text on the canvas, and then everything will be linked up with the actual publication, so that later when you come back to this map, you can again just navigate to that on a button click. Okay, then one final example here, data storytelling again. Here we're looking at a document that was uploaded in Lifelike, and let's take a look how we can extract information from a document. So here we have the ability to say, I want to create a new map. Basically, I want to maybe just take some notes as I'm reading this document. I'd like to just capture some information that I'm interested in. So you can just drag these entities that are annotated, put them on the map, and start storyboarding again and drawing and remembering information. So it's really nice and fast to do that. And everything is linked, of course, to the knowledge source where this came from. You can also highlight text and grab text. And then later on, when you actually click on this again, and let's say the document is closed, it will actually pull it up for you. I can also grab images, either from the internet, or I can just snap something from the screen. And I can also add that to the canvas. And now I can annotate this image further, and because maybe I'm interested in storyboarding that again. So you can just put some text over this to annotate it. You can also click on the on that image, and you can make it put it like a border around it, like style it up a little bit. So can I put a gray border around it and just make it look nice. You can also grab, you can always drag and drop things in Lifelike, so you can just grab that paper and drag it again over to the source, and that way it will remember that this came from that paper. So later on, when you click on it, you can open up the paper. So now we are wanting to search for this particular gene against other knowledge sources that we have in Lifelike. And let's say here we're finding, oops, I'm sorry, I apologize. So here I'm finding an Excel spreadsheet that I'm bringing over again on the map, and I'm finding another Sankey file, graph file. And so you can just grab these things and bring them to the canvas and storyboard and pull everything together. So it's a really nice drag and drop application. And all of this is stored then as basically again, as a knowledge graph. Okay. As I've already said, we do knowledge reconstruction, and you've seen it, I've demonstrated it. But basically what it allows us to do is, with this map drawing capability against the graph, you can grab any entity from the graph and you can create new associations with it, right? So this is things that you cannot go to the public databases and make updates, but if you have those public databases in your knowledge graph, you can create new relationships to continue to curate your knowledge. So what are some of the challenges that come with this? There's definitely some challenges. The benefits are great, but the challenge that we have kind of come across is really the lack of graph data literacy. So the ability to actually understand the power of graph, and being able to translate user problems into graph problems. This really is a new way of thinking. And then we need to invest in this, right? We need to raise people even out of the universities. People aren't coming readily equipped with this type of skill set. So it's really something that you have to do in your organization. You have to raise this, and you can use Neo4j and run data science boot camps, which we have done, and so on. But this is definitely something that'll take some time to get used to. Also, we cannot use out-of-the-box algorithms without considering the fit to the problem, right? So you have to first be able to understand the problem, then fit the algorithm. And so there needs to be some creativity. You need people who are creative who can do that. The lack of good visualization tools that enable us to detangle hairballs, we are working on those. We have, really been able to achieve that. It was at the beginning very, very painful. It was like we got a result back, but then we couldn't see it, and it was really painful. And then we built these capabilities, and now we can actually see things. And then limitations with NLP, the ability to extract relationship meanings. This is very hard, and if you can't do that very well, then there's only so much you can do with those. So those are some of the challenges, but I definitely, if you overcome these, then the benefits are just amazing. I think that, again, companies cannot afford not to invest in this, because it really brings surprising results. The type of things that you'd never think about. For example, that the Amazon is affecting the weather pattern in the Midwest, right? Things that you wouldn't think that are connected, you're able to connect with graph data, databases and graph algorithms. Our goal at the Center for Biosustainability is to promote these graph data science approaches and graph data literacy. We take in a lot of different students, PhD students, master students, etc. We take them out of universities, international as well as in Denmark, and then they leave again and go to other companies, international as well as in Denmark, and other universities. So there's an influx and outflux, and we want to train people and equip them with graph data science approaches. And we're already doing that in other areas, but now we're adding these graph data science approaches to that. And so we're going to be running a lot more boot camps. We've already been doing some of those, but we'll be investing a lot more into that. So, and that's the end of my presentation. I can take questions now. I hope I was able to show you the power of graph technologies and how we are supporting those with our open source platform, Lifelike.
Evelyn, thank you so much for this fantastic presentation. I think it's really, really, really impressive what you've achieved here. Yeah, and I can completely agree. We need more graph people, more graph literate people, and we should all work together to train them and get them into our projects. Let's have a look at the questions. So there are a lot of questions. Let me pick a couple of things. Let's maybe start with a question about your text mining or text analysis approach. It's a very general question, but an obvious one. How do you deal with the messiness of PDFs and the different formats and structures?
Yes, yes. PDFs are very hard to work with, and we've definitely had to put a lot of effort into it, and it's really good. It's already really good. As occasionally maybe get a paper where it doesn't annotate it well, but we've put a lot of effort into it. So and I have an expert on the team who had a lot of experience with PDF, luckily. So yeah, I think we have created something good for the world here to be able to bring in the PDFs and process them.
You created something good for the world, and you mentioned a couple of times that your platform is open source. And could you maybe go into detail with a couple of questions around the open source aspect? And is it possible to access it? Is it hosted? Can you download it? Could you elaborate on that a bit?
Yeah, so this is just happening. We've been developing Lifelike over the last about year and a half. And so it's just happening to make it open source. So it will be available very soon here. And also we are in the process of testing how scalable it is so that we can do a public deployment. So that if you didn't want to host it yourself, you can just access it publicly. And if you're comfortable, I mean, the security is there. We have, of course, permissions, project security built into it. So you can use the public deployment. So this is something we're also working towards and should be able to get that up over the next couple of months.
Mm-hmm. So it will become available as a platform as a service or software as a service even. So that is also something that a couple of people asked about.
Mm-hmm.
I think your point about educating people also resonated with the audience. So there's a question here. I do agree about that the graph way of thinking is not widespread. What would you suggest for educational programs? So how do your boot camps look like, and how do you approach that?
Mm-hmm. Yeah. So there's classes one can take in the university. But I, for example, had a great person on my team who now moved on to get his PhD, but he came out as a master's student, and his feedback was that it was not part of the curriculum. He had to choose to take classes in this area, right? And so what one can do is go choose to take classes in this area. The other one, we've hired Neo4j for running a data science bootcamp. And then we are now building up resources internally that are sort of applicable to people's problems. And then we want to run those bootcamps together with Neo4j and based on our data, so that's just kind of like what's coming up now is these bootcamps that we want to run to show them this is how you use the database we built and with the graph algorithms that we think are a better fit. And here's the tool that allows you to visualize it, etc.
Mm-hmm. Yeah. And I hope, of course, that events like our workshop here and hopefully the ongoing collaboration in this community will help to bring people with the necessary skills together.
So one more, we have not thought about opening up the data science bootcamps to the public, but we could consider that if there's an interest, actually. We thought about running it internally, but if there's an interest externally, we can also think about opening it up.
Mm-hmm. I can imagine that a lot of people would be really interested in that. So if that's possible, I think that would be fantastic for the community. Because I think especially the let's say data analysis and data science aspect is something that is that's growing a lot, and a lot of people are interested. So why not, if that works?
Okay. Yeah, I'll take it back as an idea.
Let me have a look at the questions. So there's a question about the relationship extraction and around ontologies. So how do you define your ontologies, or what kind of ontologies do you use?
Okay, we use the predefined ontologies that exist, like Cabby, Go. So mesh terms are used a lot for text mining. So we kind of use those predefined ontologies that are out there. We do not define our own at this moment.
Yeah.
But one could, of course.
Okay, so it's 5 o'clock already. I think we had a fantastic presentation to close our workshop for today. There are a couple more questions, and again, a lot obviously around open source and access and having a look at it at the platform. So Evelyn, thank you so much for your talk. Thank you so much for all the details and the storytelling about data storytelling. And I think with that, we can close for today, more or less. This was the last presentation. And Alex, what do you think?
Oh, it was really good. This was another highlight. I mean, definitely there were only highlights today, I think. But this was really interesting from Evelyn just now. I was very, very surprised, especially with the integration from like somebody said in chat as well. It's like, what do you do with the messy PDFs? And I was saying, no way. But then she showed how it's done, and it's really cool to get the information out of a messy, boring, static PDF into the graph, and then enrich your knowledge. So yeah, that was an eye opener for me.
Mm-hmm. Michael, what do you think about Lifeline?
So it was super impressive. So I especially with like 1.5 years of development time, I think that like all these aspects, so from the knowledge graph, pulling all the data sources together, the PDFs, the annotations, the integration, the querying, the visualizations, the visual storytelling, all of that is a massive software that came out of that. And I'm not sure how big Evelyn's team is, but it looks really, really impressive. And I really wish and hope that it will be publicly accessible and available as open source as well, because I think something that I've seen as a theme today as well is that many of the research groups and many of the different projects are kind of repeating the work, and everyone integrates all the data sources. Everyone does like some kind of natural language querying or path exploration, and so we see these kind of repeated things, and it would be so much better if we can, collaborate on those and don't reinvent the wheel, and rather focus more time on the actual scientific work, on like working on new types of drugs, working on understanding metabolic pathways better, or finding new ways of utilizing this data. I think it's a little bit unfortunate that so many of these of the projects need to spend so much time on infrastructure and kind of doing these things again, which I think would be amazing if you have like a baseline kind of infrastructure that people can use if they want to, and then it can really focus on the scientific and life sciences questions more as such. It would be really cool.
Mm-hmm. Yeah, I completely agree. So on the drug discovery side, we always have genes and proteins. On the medical side, we always have terminologies, disease ontologies, and all that. And if Evelyn can open her boot camps and share that. Maybe Novo Nordisk and Cepi can share their gene and protein integration.
Yeah, exactly. And then, for instance, you can imagine that a project like Health Echo could integrate with Lifelike, or Jeremy's project could integrate with this, and then it would be like at least using joint infrastructure, even if the UIs and interactions are different and so on, but at least the underlying infrastructure could be more shared as such. That would be really cool. The other thing that I found really good or interesting today is kind of just the versatility. Kind of, we've seen so many different aspects of using graphs with medical data of any kind, right? So, and it's not just like one use case or one particular type of usage that we see. We see everything from managing contracts and like patient information to clinical trials to like the document paper extractions to using, of course, lots of knowledge graphs, and then kind of what to do with this, right? So then kind of finding new cancer treatments and so on. So that's the other thing that I really find very inspiring is that there's so many things people are already doing today, but it also means that there must be so much more that's possible in the future as well, right? So I think we only see like the early beginnings of this, and if you think like, let's say 5 years from now, what will be possible there, right? So if we have all of this and more people being graph educated and being able and skilled in this, where can we be? So that's something that I'm personally very, very excited about as such.
Mm-hmm. We've seen a lot of super interesting things today, but don't forget, everyone who's still listening, that we're going to continue tomorrow, right? Exactly. So we're going to start tomorrow at 10 a.m. CET. That's pretty early in the US, but that should be okay for APAC. And we're going to have talks tomorrow about pharmaceutical graph use cases, modeling bacteria genomes in Neo4j, modeling multi-omics. So the multi-omics talk tomorrow from Clarion, I think is super interesting, also in the context of the OMOP talk today, which is going to develop into the direction of multi-omics. And later we're going to follow up with what knowledge graph analysis, knowledge graphs about drug safety, and again, more NLP, and constructing a clinical knowledge graph with NLP methods. So lots of cool stuff coming tomorrow.
Yeah, exactly. And I mean, for the folks in the US, fortunately in the YouTube live stream, you can immediately go to the beginning and kind of go back to the start and catch up from there. And so that's really nice about the live stream. So thanks a lot, Alex, for setting all of this up. Much appreciated. So today we streamed, I think, to 5 platforms or something like that. So,
you did, yeah. It was
a new record. We've never streamed to so many platforms at the same time, and just to allow people to watch it where they're interested in. And I think that's really great. And especially you can pause it, you can pick it up again, and you can speed it up if you want to watch at 2x or 1.5x or something like that. And or if you're late, then you can catch up as well. So that's a big advantage as such, which I really, really appreciate as such. So yeah, and otherwise looking forward to tomorrow. So that's really cool to see what will be coming, and then, you know, starting the collaborations. I mean, that's also kind of one of the things that we want to see, kind of the attendees, but also the presenters seeing each other's work and being inspired and being encouraged to collaborate and do things together, reach out. So don't hesitate to reach out to any of the folks presenting today, but also within each other. Please share in the chat when you're interested in collaborating. Or if you don't have the contact information, ping Alex or Martin, and we are happy to connect you as well. So that's what I also really hope to happen, that people are encouraged to, you know, start doing things together as such as well.
Fantastic.
Yeah.
Perfect.
I wouldn't know what to add to that.
Exactly. Well, thank you all for joining today. Yeah, see you all tomorrow. Take care. Have a good night or a good evening. Even morning, maybe. I don't know what time you're watching this. And then, see you tomorrow.
See you in a few hours.
See you tomorrow.